Myself, Brijan Singh, and I am assistant professor in the Department of Biochemistry. Okay, I will ask the questions, and we are just introduced in many of the classes, so don't take too much time about the introduction. Okay, so the topic of discussion today is cell, molecular and functional structure of functional organization of cell. This topic you already studied in your competitive exam when you fight the NEET exam. You already studied all the important aspects of the cell, all the aspects of the cell organelle. Understand? But question is that why it is important? Why it is important to study it in our biochemistry? Who will discover cell? One question from one. Raise your hands who know the answer. Who discovered cell? Robert Hooke. Okay. Anybody else? Anybody else? Yes? Anton Leeuwenhoek. Actually, cell discovery is not like that. Some scientists identify some things, and some scientists identify a particular cell. Robert Brown. Who can discover the nucleus? Robert Brown. Who can discover the cell? Who can discover the ATP? Lipman. I'm not going to a historical view of this that. You already know, you will read the books for that. But you understand that why it is important to study it in the biochemistry. By the discovery of electron microscope, we identified that something is present in the cell, such like as a physics. In the physics, what we have? We have atoms. In these atoms, there is electron, proton, neutrons are present. Understand? Same is in our this biochemistry cell in our biology. The physical existence is occur by the cell. And in the cell, there is some cell organelles are present. It is like that, electron, proton and neutron. Students, don't do that. Don't rotate the paper in my class. First of all, you listen carefully. While I will taking the class, what I expect from you? I expect nobody disturb the class. Be attentive, listening what I say. Understand? I will ask the questions to you randomly. So don't rotate the paper, sir, this is your first day. Understand? And you know, first expression is the last expression. Understand? So many of the things we discuss upcoming classes, why are you take interest in this MBBS course and what is the motto of this course. We already discussed all the things in next classes. Okay. But today our motto is we are come here to take the knowledge. Okay, why are you came here? Why are you wake up in the early morning and came here in the class? To take the knowledge, na? To take the knowledge. So why are you just, don't rotate the paper for attendance, they will take the attendance and somebody is taking your attendance. So don't worry about that. This is being your first class of the sessions. Okay, be attentive in the class. If something is not understood by you, so you will ask after the class. Okay. So, the cell is what? Cell is important like that, what I say. It is like a physics, that is atoms is particularly smallest organizations of particular things. Same is in our biology, cell is a structural and functional unit of life. If we go to the definition of the cell, what is that? It is a structural and functional unit of life. Okay. But when we talk about the biochemistry, why it is important to study it in the biochemistry? Because in the biochemistry, you already know, if you know the definition of biochemistry, you are able to understand why it is important to study the cell in the biochemistry. My question is to all of you, what is biochemistry? Just raise your hands who know. If he or she attend the class, before attending the class of sir, raise your hands, what do you expect, what do you know about the biochemistry, what do you think about the biochemistry, what is biochemistry? Raise your hands. Yes. Nice answer. Anybody else who can give more clarification? Okay. Biochemistry is basically a science which deals with the study of chemistry, chemical reaction proceed in a living cell, proceeding in the living organism. There are many types. You already know the many of the things. You already know the TCA cycle. I think many of them know. You know urea cycle. Huh? You already know that. So simple, if we going to discuss about the cell, the first thing is that structural and functional organization of the cell. Structure and function organization of the cell, and it consists what? It consists of membrane. It consists of cellular organelles. And these cellular organelles contains many of the enzymes. Enzymes, you know, that's something, the factor which can regulate the chemical reaction. Okay, which can regulate the chemical reactions. Either it can increase the reactions or decrease the reactions. We discuss in the next class about the enzymes. So, what is that? The cell which can have some organelles, then can proceed some of the metabolic pathways, some of the reactions of the metabolic pathway of biochemistry, such like that glycolysis. Glycolysis. What is that glycolysis? I think you already know, but glycolysis is something that is the breakdown of the carbohydrate molecules. It is okay, it is like a glycolysis. But it is mainly proceed in what sections? It is proceed in cytoplasm and mitochondria. Cytoplasm and mitochondria. That is ETC, electron transport chain. It is always proceed in mitochondria. How the ATP is produced? It is proceed in mitochondria. Okay, urea cycle. It is proceed in cytoplasm and mitochondria. Cytoplasm and mitochondria. Okay, and your protein synthesis. That's you know a word that describes the protein synthesis, that is central dogma. Central dogma, that is protein synthesis. It is mainly occur inside the cell in cytoplasm. In which unit? Ribosomal unit. There is insertion, cutting, packaging of the protein molecules all proceed in which important suborganelle? That is cell. So that's why it is important to study the cell in our biochemistry. First thing. Okay? Then many of the reactions of the metabolic pathway in the proceed in different compartment of the cell because why it is happening in mitochondria? if I will say glycolysis happens in cytoplasm and mitochondria. Why? Because all the markers enzymes which are present in the glycolysis are present in cytoplasm and mitochondria. That's why it is important to study about the cell in our biochemistry. Okay? There are many of the reactions, many of the processing of the cell, many of the things. Protein synthesis are mainly occur in the cytoplasm, are mainly occur in the cell. That's why it is need to be important to study about the cell. Okay? So in this cell section, what we are going to study, we are studied about the eukaryotic cells, prokaryotic cells. I think you already know about the eukaryotes and prokaryotes. Huh? If you being qualified in the NEET exam and getting the rank, I'm not telling about the difference between eukaryotic and prokaryotes. You already know it is pre-existing and new one. Okay? So, we discuss about the difference between prokaryotes and eukaryotes, some features of eukaryotic cells, cell membrane, plasma membrane, and the models which can describe the structure of the membrane. Okay? So, the first thing is that when we are going to discuss that yes, this is the general diagram. You will see the general diagram of the cells, that it contains cell membrane, lysosomes, mitochondria, Golgi apparatus, ribosomes, peroxisomes, intermediate filaments, RER, rough endoplasmic reticulum, smooth endoplasmic reticulum, cilium, and cytoplasm. Students, these all the organelles are very important because these all the organelles can synthesize some kind of protein, can have some kind of marker enzymes which can proceed different pathways. Understand? And your examination point of view, it is also important. So what is cell? Cell is a structure and functional unit of life. By the cells, group of cell forming tissues, and group of tissues forming organ systems. So it is important now. Every single child, just like that, you are sitting in a group, you all have important for the counting. Understand? If I am not counting a single student, the counting being disturbed. Just like that, you expect all the cells making the group of tissues, and these group of tissues making the organ systems, and these organ systems performing a living system. So it may be a unicellular, multicellular. We discussed both of things. Understand? So what is mainly aspect? That is the nucleus. And in this nucleus, there is the presence of nucleus, the fluidy material inside the cell. Okay, so all the things. See, cell change karo. Okay, so what we are going to discuss, the classification. Just एक काम करो, इसको यही दे जाओ, इसको यही दे जाओ कीबोर्ड को. इसकी सेल नहीं है क्या? What we are discussed, classify living cells, molecular and functional organization of the cell, and structure and function of cytoskeleton. Cytoskeleton means how the cell's structure is formed and what kind of proteins, microfilaments, microfibrils are present to make the structure more stable. Okay? To structure more stable and describe the cell fractions and marker enzymes for different organelles. So this is being the process through which we can separate out our cell organelle on the basis of density. That is called cell fractions. Understand? If we adding such like that in a solutions NaCl, sugar and some kind of more density urea powder. If we are going for fractionization, then it is separate out on the basis of density. Understand? So it is called cell fractions. Now, this is a general diagram. What is cell? If somebody is saying that is a cell is a structure and function unit of life that contains many organelles, mitochondria, Golgi apparatus, and lysosomes, vacuoles, many of the things. Okay, we discuss later. Archaea living in extreme environment and this kind of stuff, unicellular organelles. Firstly, we discuss about the eukaryotes and prokaryotes. Okay. Primitive types of cell is known as prokaryotes. New one types of cell is known as eukaryotes. But it is not easily defined like that. There is a difference. Difference of membranes. Prokaryotes not having a membrane, but eukaryotes have the membrane. Okay? Simply, the difference is on the basis of the membrane, that is eukaryotic cells. It contains nucleus, which is also a true nucleus. Why it is called true nucleus? Because it is surrounding by the membranes. Okay? So it is make the differences. Now, what are the difference? This is a very important slides to all of you about the university examinations also. The main question is asking in the exam, what is difference between eukaryotes and prokaryotes? Okay? So it is organelles, eukaryotes, nucleus is present, but in prokaryotes not defined nucleus. Not defined nucleus means there is not a particular membranes are present. All of you understand English? How many students from Bangla boards? Oh, many of us. You are capable to understand the English? Hindi mein chalega. Because I am not speaking Bengali. You know? Okay. Because I am not from this state, I am from Madhya Pradesh. Madhya Pradesh, Gwalior. Okay. So now we go to a topic, no defined nuclear, DNA present but not separated from rest of the cells, plasma membrane present, mitochondria present, endoplasmic reticulum present, and these organelles, all the organelles are present in prokaryotes and eukaryotes. But when the question is asking, you have to write basically a 5 difference between them. Okay, that is the process of separate out from each other is mitosis or by binary diffusions. Viruses, the viruses is a not living organism that in the sense of the cells and it is capable to replicate. You already know the virus. Why? Because we are just across the epidemic criteria, that is COVID-19. So I think you already know what is viruses. That is something which can replicate itself, make the several copies and make the infectious human beings infectious. Okay. Now we directly go for some molecular and functional organization of cell and its subcellular components. Now students, if we are going for the study of the cell, how can we study it? And what is present in your syllabus? What is basically you are going through the study of this cell? That is, it contains cell, cell organelle, first thing. Second one, cell membrane. Protoplasm. endoplasm or be called as nucleoplasm. Now in the cell membrane we can separate out our this cell membrane as single membrane cell structure and double membrane cell organelle. Double membrane, sorry, this is not a structure, this is organelle. Okay, organelle, and this is double membrane cell organelle. Which one is double membrane cell organelle? Mitochondria. Very good. Mitochondria. Mitochondria is very important. And rest of the organelles are considered to be a single membrane structure. Okay. Rest of them are just like this: ribosomes, lysosomes. Don't talk. Miscellaneous. Endoplasmic reticulum. So major of the portions are cell organelles. That is, it has a three major component. The first one is plasma membrane, cytoplasm and nucleus. We already know. So first thing is that we are discussing firstly the membrane. So students, why it is membrane is important? Protection from what? You are talking about the cell. I am not talking about the tissue system. I am talking about the cell. Okay, next. Next. Yes, that is important. Cell is what? Cell is outer membrane of the structure, and due to the property of the cell, it is maybe a hydrophilic or hydrophobic. First thing. Second one is it how it is depend, how it take the component inside the cell. There are several processes you already know: pineocytosis, phagocytosis. Okay, all are preceded by the cell membrane. First thing. Okay, there is facilitated diffusion, active transport, passive transport. So transport mechanism you studied in the next class, either in the biochemistry or in the physiology. Okay, because your syllabus is changed. So cell is what? It is the outer layer membrane of the structure, which is primarily composition of lipids and protein. The basic structure of cell membrane is lipid bilayer. Large globular proteins molecules are interspread in this lipid bilayer. Now we are talking discuss about the cell membrane. If we are going to discuss about the composition of the cell membrane, what it have? It have proteins, it have lipids. Proteins, you know. Just a minute. Proteins, you know. Proteins, what kind of protein you know? You will eat egg. अंडे खाते हो? मैस में अंडा ही मिलता है ज्यादातर. डीम भात. That is the contains proteins. Okay, and lipid. Lipid, you know, triglyceride, cholesterol. कितनी बड़ी-बड़ी चीजें हैं. इन सब के बारे में हमें पढ़ना है साल भर. बहुत इंपॉर्टेंट है, बहुत इंटरेस्टिंग है, और इंटरेस्ट के साथ में पढ़ेंगे देन यू विल गेट लॉट्स ऑफ नॉलेज. ठीक है. ड्यूरिंग द क्लास नो टेंशन, कोई टेंशन लेने की जरूरत नहीं है, बिल्कुल बेफिक्र. कोई चीज समझ में नहीं आती है, यू विल आस्क टू मी सेवरल टाइम्स, डोंट वरी अबाउट दैट. ओके. सेल बाकी अदर फैकल्टीज अदर टॉपिक लेंगे, सो नॉट वी आर. ओके. So if we talk about the composition of the cell membrane, that it contains lipids and protein in a different proportionate, and the function of the protein is different. It is participate in the transport mechanism, it is participate in the different cellular process like phagocytosis and pineocytosis. Okay, it is mainly important for the transportation of sodium and calciums. Okay, and if we talk about the shape of the cell, it is amoeboid shape, that is irregular shape. Okay, but when we talk about the body cells, neuron cells, muscle cells, they have in different shapes. Understand? And for membrane, if we talk about the muscles, it is called sarcolemma, that is called the outer membrane of the cells. So different membranes to be called at different organ systems, the membranes to be different types. Okay. So this is the general structure of the cell membrane, that it contains hydrophilic end and hydrophobic tails, carbohydrate moieties, peripheral proteins, lipid barriers, and integral proteins. Understand? So students, before going to understand what is the meaning of intrinsic protein, what is the meaning of extrinsic protein, the protein which is present outside the cell membrane, just like that, this is your cell membrane and this is the protein, and we call this protein as extrinsic protein. Okay? And if the proteins are present inside the cell membrane, it is called intrinsic protein. Okay? Some like that, the structure, you know, some kind of, you will see there is, what is that? Comb-like structure you will see, red color, pointer. ये पॉइंटर वॉइंटर तैयार करके रखो करो भाई, बीच क्लास में बोलना ना पड़े. That is, you know, this is the lipids. This is the lipid molecules. Students, in our cell membrane, there are several types of lipid membranes are present. It is complex lipids are present. Complex lipids are present. Okay, it is phospholipids. It is phospholipid. The phosphoric acid are attached to the lipid bilayer. Okay, this is lipids. And with the interaction of the water, it can divide it as hydrophilic and hydrophobic ends. Can you define anyone? The second one. What is the meaning of hydrophilic end and hydrophobic ends? Okay. Okay, non-polar. Yes. So the membrane is sometimes referred as fluid mosaic model. The membrane mosaic is fluid because most of the interaction among its components are non-covalent, leaving individual lipids and proteins molecule free from the move laterally in the plane of the membrane. So this is the structure, general structure of the biological membrane. It contains integral protein inside the cells, carbohydrate moieties, peripheral proteins, and glycolipid, phospholipid, sphingolipids, different types of lipids. Okay. Now this is the general structure, very important structure. When the question is asking, write about the fluid mosaic model. Question is asked in the university exam. What is fluid mosaic model? Okay. So you will make this diagram and elaborate it point wise in your formula. That is fluid mosaic models. You already know that it contains carbohydrate moiety, it contains cholesterols, glycoproteins. What is glycoprotein? Protein plus carbohydrate. It is not like that a mathematics, protein plus 2 plus 10. It is not like that. It is a conjugated protein. It is a complex protein that contains carbohydrate moiety attachment with the proteins molecules. Give your answer with your clarification. Okay? Hydrophilic region, hydrophobic region, phospholipids. Why it is called phospholipids? Because it contains phosphoric acid at the top of these lipid molecules. Integral proteins, peripheral proteins. Okay, this is called fluid mosaic model. Okay, who can discuss fluid mosaic model? Good. Singer and Nicholson. Singer and Nicholson describe the cell membrane structure, that it contains carbohydrate body in there. They are kinds of different proteins, which is to be called integral protein or extrinsic protein. We discuss detail. The lipid of the membrane provides a barrier that obstructs the movement of the water and water soluble substance from one cell compartment to the another. That means it's being helpful for the co-transportation and transportation of the molecules. Okay? Proteins molecule in the membrane after do not penetrate. This is actually the process of osmosis and diffusions occur on the basis of cellular gradients, cellular densities. Okay? Many other membrane proteins are enzymes that catalyze different types of chemical reactions. Proteins molecule in the membrane do not penetrate all the way through the membrane organized by axial force for passage of a specific substance through the membranes. So this is the compositions. Write in your copy. Don't see my face. Understand? What do you think? Your lecture is over. If you are not going to study it after the lecture, this is a skip out from your brain. You, what happened? Pen nahi hai, copy nahi hai. So the composition of the cell membrane, it contains 55% protein, it contains phospholipid 25%, and rather than other molecules, cholesterol, lipids, carbohydrates. Okay? So this is the composition of the fluid mosaic model. This is the composition of cell membrane, and in this lipid membranes, it contains two types of lipid we call as, one is hydrophilic, because each lipid phospholipid molecule is soluble in water, that is called hydrophilic, and the other end is hydrophobic. This is called tail group. What is the meaning of head group and tail group? Such like that, in the cell membrane, these kinds of lipids are present. This is the top. This is the phosphoric acid which is attached with the lipid bilayer. Okay, so this is called head group and this is called tail group. The phosphate end of phospholipids is hydrophilic and free fatty acids. Okay, understand, students? What is that? What is the attachment of this phosphoric acid? This is called FFA. That means free fatty acids. One tail free fatty acid, two tail free fatty acid. And it is important. Not a single pathway is proceed. When we are going to discuss about the prostaglandin, when we are going to discuss about the prostaglandin synthesis, arachidonic acids. Okay? So these are important. So basically in the cells there is a phosphoric acid is attached with two free fatty acid chain. Okay? Now you will see the structure, that is free fatty acid chain structure of phospholipid, a common glycerophospholipids and other one is phospholipids, that it contain aliphatic side chain. Aliphatic, you know, you already discussed, you already studied from the organic chemistry. What the meaning of aliphatic? Alkaline containing long chain. Understand? Hydrophobic tail group, fatty acids, phosphoric acid and alcohol. So its may be changed. Alcohol group is changed by sphingosine and sphingosine, sphingle is to be called. When we discuss about the lipids, you already know about all these things. Okay. So the principal phospholipid in the membranes are glycerophospholipids, sphingospholipids. What is the meaning of that? The alcoholic group which is present in the phospholipid is glycerol. Okay. And in the sphingomyelin, it is present in sphingol. The alcohol group is present in the sphingomyelin is sphingol. It is present in not in, if we talk, it is present in the white and grey matter of the brain. Understand? So it, from the small things you learn large thing. That means from the structure of the membrane you understand what is present in the grey matter, what is present in the white matter of the brain. Understand? So it contains phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, several types of membrane lipids are present, and this lipids composition varies different cell types. Okay? Cell membrane, for example, it is cardiolipins because it contains mole cholesterol. Mitochondrial membrane is very low in cholesterol, sphingolipids that contain cardiolipins. What is this cardiolipins? That is, cardiolipin is what? The lipid molecule which is present in the heart cell, cardiac cell. It is called cardiolipins. Okay? Now this is the structure of the plasma membrane structure components, fluid mosaic models. Okay? We already discussed about all these things. That is glycoproteins, globular proteins are present. Achha, why it is called globular proteins? Globular proteins is nothing. This is like the shape of the protein. On the basis of the shape of the protein, it can divide it. It can divide flatel protein, spindle shaped proteins, globular proteins like myoglobin, albumin. Okay? So don't confuse rather than. It is globular proteins, cholesterols are present, phospholipid bilayer are present, integral membranes proteins are present. Okay? These are some receptors proteins are present. Why it is important receptor proteins? To recept a particular molecule. And it is important for signal transduction. Okay? So membrane is important for signal transduction process. Just like that, when an insulin is released from the body and it is going inside the cell to proceed this process called glycolysis. How it is proceed? How it is enter in the cell? By the recognition of particular receptors. आप इस क्लास में कैसे आए? कैसे पता पड़ा? There is a marking of LT1. पता था? Same like that, in the cellular membrane, if some molecules are enter in the cell, it is by the process of these receptor proteins. By the identification of these receptor proteins, several molecules are proceeded. I am not going through deeply on this topic. We discuss later. Okay. You just understand what the meaning of these receptor proteins. Why it is important? Is it important for signal transduction process? Okay. Now the cholesterol. Okay. Cholesterol, what the cholesterol is very bad molecules in the biochemistry aspects, in the medicine aspects, and to treat the patient, to treat the subjective patient, cholesterol is very bad. Okay. So the cholesterol molecule in the membrane is also called lipid in nature. Why it is important? It important for maintenance membrane fluidity. Membrane fluidity. Unsaturated fatty acids, chain of glycerolipids bend to cis conformation for pockets of cholesterol which bind to the hydroxyl group in the external hydrophilic region in the membrane and hydrophobic steroid molecule nucleus, hydrophobic cores. Basically, you are not go through that. You are just understand there is the presence of cholesterol, there is the presence of some free fatty acids, some phosphoric acids, and on the basis of their conformation, they have two types of conformation, band cis conformation and trans conformations. Cis conformation and trans conformations. Okay. On the basis of this separation, there several cholesterol molecules are present. The presence of cholesterol, the cis unsaturated fatty acid in the membrane prevent the hydrophobic chains from packing too closely together. how the molecule becomes saturated and unsaturated? On the presence of bond. If it is single bonded, it is unsaturated. If it is, it is single bonded, it is saturated. If it is double bond, it is unsaturated. It is not dissolved. Okay. So on the basis of these bonds, it can make more complicated hydrophobic chains for not entering outer the molecules. It is become for making the property of this membrane hydrophobic, water repellent. Okay. The change in the membrane fluidity may affect protein and span of the membrane. Ion channel receptor for neurotransmitter involved conducting the nerve impulse. It is basically for the taking of calcium, potassium for the nerve conduction. Okay. The cell membrane, which is also present in the muscles, it is also present in the muscles, in the neurons, sarcolemma. So what is that? This cholesterol became taking this sodium and potassium, okay, and this cholesterol become helpful for conduction of this nerve impulse. Now membrane proteins, there are two types of proteins you already know, integral protein and external protein, and on the basis of their composition, it is divided as simple protein, complex protein and derived proteins. But here, this is complex proteins, glycoprotein, which is a combination of carbohydrate and the proteins. Integral protein membrane that protruding all the way through the membrane. What is the meaning of protruding? Protruding मतलब होता है निकला हुआ. Such like protruding belly, किसी का पेट निकला हुआ है. ठीक है, protruding belly किसी symptoms में किसी बच्चे को देख रहे हैं, there is a clinical symptoms of protruding belly. That means there is large enlargement of the liver. Such like in our cell membrane, अगर किसी cell membrane से कोई protein molecule बाहर निकल रहा है, it is called protruding proteins. Peripheral membrane protein. Peripheral proteins are attached only one surface of the membrane, do not penetrate all the ways. So now function. What is the function of this membrane? Integral membrane proteins primarily as channels through which water molecules and water soluble substance, especially ion, can diffuse between extracellular and intercellular fluids. That is called ECM, extracellular fluids and intercellular fluids. Means on the basis of recognition of these integral proteins, these calcium ions by different transport mechanisms, it can enter in the cell and perform their function. Okay. Now, these are act as a receptors for hormones and neurotransmitters. You already know. I discuss here. That is signal transduction. Signal transduction is what? Identify of receptors. Okay. Peripheral proteins function almost entirely as enzymes and its controls of transports of substance through the cell membranes pore. So basically, if we see the whole function of this protein, it acts as a receptors, first thing. It acts as an enzyme, second thing. It acts as a taking of different substance from extracellular fluids to intercellular fluids. Okay. Membranes, carbohydrates, what kind of carbohydrates are present? They are glycoproteins, that is conjugated proteins, carbohydrates are present. Some proteins and lipids are external surface of the membrane, short chain of carbohydrate we called as oligosaccharides. In the next class, there is some faculty taking, main faculty taking the chemistry of carbohydrate. On this class, you understand what the meaning of oligosaccharides, what the meaning of polysaccharides. You already know well, but on the basis of number of saccharides units, we can divide it. Okay. There is proteoglycans which is present loosely from the cell membranes, a carbohydrate motif which is present, we called here in the cellular membrane structure, it is called proteoglycans. Where is proteoglycans here? Where is proteoglycans here? These are the proteoglycans attached with the cellular membranes. Okay? These are the proteoglycans. What are you doing? हाँ कुछ बता रहे हैं. अभी चल रहे हैं उसी पे आ रहे हैं. अभी आए कहाँ वहाँ पे? We come here, don't worry. We come here to the next slide. We discuss that. Okay. So, as many other proteoglycans are loosely attached to the outer surface of the membrane, the entire outside surface of the cell has a loose carbohydrate coat. It is called glycocalyx. Okay. What is that? That a cell membrane, what we call outer protein molecules of the layer we call as extrinsic proteins. Okay. But in the case of carbohydrate, when it is attached with the membrane, it is called glycocalyx. Okay. There is an attachment of the carbohydrate, different carbohydrate moiety to the membrane. It is to be called glycocalyxes. Okay. Now what is the function of this carbohydrate? These all are the functions that many of the carbohydrate had negative electric charge, which giving most cells an overall negative charge that repels the negative object and restrict the effect of hydrophobic compounds. That means it may be helpful for co-transportations of the ions and molecules by the electrostatic charges, ionic bond interactions. The glycocalyx is some cell attaches to the glycocalyx of the other cell, that attaching cells to one another. Now you see the diagram first, because if you are not seeing the diagram, I think you are not able to understand what is the meaning of glycocalyx. These are the molecules which is to be called glycocalyx. Understand? The carbohydrate moiety which is attached to the membrane, outer membrane. What is this red spot? Anybody else? This is phosphoric acid. This is free fatty acid. Why it is called lipid bilayer? Because lipid are present over like that. Huh? And this is proteins are present. Students, when the question is asked, you have to make this diagram very clearly in your mind and elaborate it. Okay, so now for the next class we all discuss about the cytoplasm and its organelle in detail, about the mitochondria, about endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes and peroxisomes. Okay, so what we are discussed today, we discuss fluid mosaic model. Okay. Function of the cell membrane, composition of the cell membrane, lipid bilayer model. This is called lipid bilayer. Now I ask the question, who can maintain the integrity of the cell membrane? Which composition, which chemical constant making the integrity of the cell membrane? Fluid integrity of the cell membrane. Cholesterol. Okay students, so next class we discuss all these cell organelles and others. ठीक है.
हाँ भालो भालो लागले भालो. So why we should study blood? Have you any idea about blood? What helps us blood? কি সাহায্য করে? Yes, transport, very good. Anyone? Provide? Nutrition provide করে না, nutrition কে carry করে. Then transport of what? কি transport? Heat transmission করে, yes. Body temperature regulations and transport of oxygen, transport of carbon dioxide out of our body. The waste products carry some hormones, carry some important cells. তাই তো 10-15 একদম যা বন্ধ করে দেবে, ঠিক আছে? So blood components means have to know as a clinician, as a doctor, what is the importance of blood and why I should know about the components of blood. That's very important. আমি যদি না জানি যে এটা আমার ভবিষ্যতে কি লাগবে, there is no importance of this class. Suppose a patient come with pallor. Do you know the term pallor? Pallor means ফ্যাকাসে লাগছে, সাদা, হ্যাঁ, রক্ত কমে গেছে, তাকে আমরা আমাদের মেডিকেল টার্মে বলি প্যালো। ওকে, নাম্বার 1। নাম্বার 2, সোয়েলিং অফ ফেস, লেগস, পা মুখ ফুলে গেছে, উইথ ফিভার, জ্বর আছে। Then come to your chamber or any clinic. Then what should I do? First of all, I am not going details about the how to approach a patient because this is not a time. But I am concerned about the blood, why such patients need the blood, because we recommended that some investigations. Investigation means complete blood count and also CRP, C-reactive protein, and complete blood count is must for each and every patients to look any infection is there, which type of infection is this. Blood components এর মধ্যে pallor means আমি hemoglobin level আমাকে দেখতে হবে, so we should know the hemoglobins. In the complete blood count there is a lots of test is done. Complete blood count not a single test, is a series of tests like hemoglobin, total count of WBC, total count of RBC, then MCV, and you will come across the term. ধীরে ধীরে তোমরা এগুলো জানতে পারবে। MCV, MCH, MCHC, PCV means fat cell volume. So these are coming in this complete blood count. And CRP detects whether there is any inflammation is not. Whether patients is having some inflammation, fever, that means there is some inflammation or infection is there. CRP is a detector, is a acute phase proteins, and this called as a C-reactive protein. That is produced by the liver, and it comes only when there is some inflammatory process. আমার একটা পেশেন্টে যদি কোনো inflammatory process হয়, then only that will rise. যেমন ডেঙ্গুর সময় আমরা এই দুটো টেস্ট সবসময় করি to see both whether it is a viral component and CRP influence is there. কারো যারা পেশেন্ট যারা খারাপ হয়েছে, so their CRP is more than 100, 102, but it is less than 6 is a normal level. I am not going details into the normal levels now, but why we should know the components of blood is very important, because we have to detect with some investigations, and that is the blood. Now, if you collect a blood in a, suppose in a syringe, and then transfer into a test tube, and it set up in the centrifugation machines. Centrifugation means rotating, speed rotations. Some time is there. It will be practically, you will see in my practical classes. But if we collect the blood, then suppose there was initially there's this pool of blood is there, but after the centrifugations there is some separations. Which separation will be there? কেউ বললি একজন সিরাম, কেউ বললি একজন প্লাজমা. So that should be clear in this class. What do you mean by serum and what do you mean by plasma? These are not same. Simple formula সারা জীবনের জন্য মনে রাখবি SPC means yes, plasma minus clotting factors is equal to serum. That means when we centrifuge the whole blood and separate it into 2 parts or 3 parts, that is along with the clotting factors, it is plasma. But when we centrifuge the clotted blood and then the separation, then will this come as a without clotting factors. That means serum is without clotting factors and plasma is with clotting factors. তাই তো ক্লিয়ার হয়েছে? আর কখনো জীবনে ভুল করবি না, দুটোকে কিন্তু অনেক আনফরচুনেটলি অনেক ডাক্তারবাবুরা এই কনসেপ্টটা নেই। দরকার হয় না, পেশেন্ট দেখার ক্ষেত্রে হয়তো দরকার হবে না, বাট দরকার হবে when you will write in your prescription that we need serum calcium level. Suppose a patient is coming with some convulsions or some tetany-like movement that is due to the calcium level loss, suspecting, and so we prescribe that the serum calcium, not we are giving the plasma calcium, no. So serum is plasma minus clotting factors. Without serum is without clotting factors. Clear? Now, after the centrifugations, what will happen? Separations of mainly 3 parts. তাই তো 3 parts এর মধ্যে কি কি থাকবে? এটা একটা প্লাজমা থাকবে, এখানে কি থাকবে? মানে আমার যত রকমের ভারী dense density particles will precipitate in the lower level and low density will go up, and in the between there is a buffy coat. It is called buffy coat, and this is basically a blood. This one is a RBC. Okay, here RBC is present mainly, and here it is present কি কি থাকে? Anyone? প্লাজমাতে কি কি থাকে? কি আবার সিরাম করছি? আমি প্লাজমা নিয়ে কথা বলছি। ওয়াটার, কত পার্সেন্ট ওয়াটার? হ্যাঁ, দেখ তোর দারুণ সুন্দর তোরা প্রশ্ন করছিস। গুড, মোর দ্যান 91 or 92 যাই বল, 95 কোনো বই লেখা আছে, মোর দ্যান 90 is water. Then solutes. In the solutes, bol plasma proteins, okay, then nutrient, yes, nutrient is there, yes, then electrolytes, yes, very good. Now within the electrolyte, which one is a predominant? Sodium, calcium. Electrolytes is not a glucose. Glucose is not a electrolyte. Electrolytes means sodium, calcium, potassiums. These are. So this sodium level is high in the plasma. Sodium level is high in the plasma. Then calcium. Then some anions will be there. So glucose, lipid, fats also will be there. Nutrient as a nutrient. Kyu ekta bolli na tora nutrient. So these things are happening in the plasma, not serum. For percentage is a 50 and 5. Eta 45, তাহলে তো হয়ে গেল 100. Now it is less than 1%. Now what about the buffy coat? Buffy coat is platelets, then WBC. আর কিছু? No. Now what do you mean by platelet? Platelet is a cell. Platelet কিভাবে আসে? Is platelet is a cell? No. This is the fragment of cell. Yes, this is a part of megakaryocytes only. Megakaryocyte is a huge, and suppose this part will come to circulation as a platelet. So remember this, platelet is not a definite cell, it is a fragmented part of the megakaryocytes. It's very important and written and asked is good. Professors can ask as a in your viva also. So up to this, when we are dealing a patient having with some fever, some lack of blood, or some history of any types of accident, any types of trauma, that means some blood loss is there. So we have to do some investigation whether patient is suffering from any types of infection in case of infected patients or any type of hemoglobin loss in case of traumatic patient. So whatever it may be, it is going to have to do the CBC. If we suspect the patient is suffering from some infection, then we detect the CRP level. CRP is a acute phase protein, that is C-reactive protein. It is a protein. It is basically present in the in case of infection, it into the blood. It present is the level is high. Viral fever CRP level increase of fungal level increase of parasitic infection, bacterial infection, whatever it infection. Infection means we know that is some parasites, that may be viral, that may be fungal, that may be bacterial. We have to detect because in your mind, because we have to treat that patient. If we think that is a viral, so there is no treatment of it. Dengue kono treatment নেই যে আমি একটা ওষুধ লিখে দেব। No. কিছু কিছু ভাইরাসের কিছু কিছু ট্রিটমেন্ট হয়, যেমন হচ্ছে একটা হারপিস জস্টার। হারপিস জস্টার শুনেছিস কেউ? আস্তে আস্তে শুনবি। This is your first class. So in that case some antiviral is present. Gingival stomatitis. That's also a viral. That is also due to the herpes. So herpes, in case of we use the acyclovir, this is the antiviral drug. Remember always that mostly most fevers or most is coming now is in this fever in your family also. Some is sort fever and low cough is there. কেউ বলছে আমার abdominal pain is there. To some extent that may complain as a loose motions. That means some multiple problems are there. These are mostly, believe me, that is more than 95% cases is a viral. That means you can assure that patients that the viral load is primarily is high, but after some time or some days, that is a 5 to 7 days, viral load or viral replication will be decreased, and so on. The temperature, everything will be subsided down gradually. No need of any antibiotic. If you use antibiotic at this stage, that means it is irrational. If not focus, যে তুই কোনো কিছু বুঝতে পারছিস না, আমার এখানে একটা পাস হয়ে আছে, in that case we only use the test and the CBC will detect the whether it is viral or not. Because in case of viral picture, neutrophil count will be low and lymphocyte count will be high. This is a viral picture, and also as well as only typhoid fever may present as a neutropenia with relative lymphocytosis. কথাগুলো একটু অভ্যাস করতে হবে আস্তে আস্তে। So any types of infection we detect to see the level of neutrophil, because neutrophil is our defense mechanism. Defense mechanism means it will come to attack. এই যে আমাদের অনেক সময় হয় না যে একটা জায়গাতে ইনফেকশন হলো নিয়ে পাস বেরোচ্ছে, হয় না? পাস বেরোচ্ছে, আমরা পুঁজ বলি বাংলায়। So it's coming out why? Because the involvement of the neutrophil. So that's the great thing. That is the great thing that our defense system is active to protect or to engulf the organism that is invited in our body to attack us. That is a important clue. That means in that case neutrophil will be more than 70%. You already don't know that at this stage that what is the level of neutrophil, etc., etc. Do you know which type of cells are present over all leukocytes? WBC এর মধ্যে কি কি থাকে বলে ফেল। Yes. Never let monkey eat banana. Yes. আর জীবনে বলবি কোনো কিছু বলা যায় না, এগুলো আবার তোদের আসবে পরে। নিউট্রোফিল, লিম্ফোসাইটস, মনোসাইট, ইওসিনোফিল এন্ড বেসোফিলস, এর মধ্যে কিছু কিছু গ্রানুলোসাইট আছে, অ্যাগ্রানুলোসাইটস আছে। So these things are very important. Each and every point is important because this neutrophil is for the infections is may high. Lymphocytes is also may high in infection, and also specifically in case of any tuberculosis patients, lymphocytes level, lymphoma, suffering from lymphoma or tuberculosis, TB, যক্ষা বলি আমরা. In that case, lymphocyte level will be high. Monocyte is specific. If it is patient is coming with monocyte is more than 10%, 5%, that means the patient may suffering from any types of blood cancer. So eosinophil it is allergic. Basophil also is too much, very, very less. Okay, so these things are coming day by day when we go across this. The components of blood basically this basically এখান থেকে নিউ কি আমরা শিখলাম. আমরা শিখলাম যে আমরা প্লাজমা আর সিরামকে আলাদা করতে পারবো. Serum means plasma minus clotting factor. That means there is no any types of clotting factor in serum. But in case of plasma, এই যে প্লাজমার কম্পোনেন্টস এর মধ্যে আমরা কিন্তু ক্লটিং ফ্যাক্টরস বলতে পারি. আর কি শিখলাম? Centrifugations করা হলো, then separation on 3 layers. High density is common level is 45% is RBC. And coffee coat is presentations of the leukocytes and the platelets. Siglum, we learned that the platelets is not a definite cell, it is a part of the megakaryocyte fragmented part. Megakaryocyte থেকে fragmentation way that comes as a platelet. And platelet is a hugely, you know already, the platelet কি কাজ? Blood clotting, okay, very good. And then this is the more than 91% having water, solutes, mostly this is a sodium, and also some calcium is there, nutrients like glucose, carbohydrate, fats are there, hormones also there, hormones are there, cations, anions also there, and everything is there in case of plasma. So whenever you are writing to detect any types of anions or cations or electrolytes, we prescribe or we write that that is a serum calcium level, serum sodium level. We approach this. So you keep in mind that it is serum means without clotting factors. Okay. Now, one of the important part of your clinical scenario is that whenever we are dealing with a patient like simple fever, like now in your family also, but itself you yourself is suffering from some sorts of cough, some irritations in the throat is coming through. This temperature is favorable for any types of virus. Any types of virus, that may be adenovirus, that may be coronavirus, that may be rhinovirus. Little child, that's my domain, is suffering from respiratory syncytial virus, RSV, is coming with acute bronchiolitis. So these patients, whatever it may be, if its persistent fever is more than 5 days fever, and already I told you that a 5 to 7 days fever is normal in case of viral fever. But still, if it is more than 5 days and the patient is having high fever, then we have to prescribe or have to write down some investigation definitely to save the child and to save us also. আমি মনে করি কিছু করলাম না, আমি বিশাল কিছু হয়ে গেছি, না কিছু নেই চলে যাও, then she or he may turn into the sepsis due to my negligence. So that is the beauty of this investigation. If you simply do a CBC or CRP, it's not so much cost. If it is more than 5 days, I am not telling you at all that you first time you are coming the patient and first day you are writing the investigation. If not indicated properly, then you have to wait. Remember always, in case of any types of viral infections, mostly common viral infections, patient will be absolutely fine during the febrile stage. Febrile means যখন জ্বর নেই, এটা আমরা জিজ্ঞেস করি. But whenever he or she is suffering, that may be child, may be adult, some uneasiness he or she feel. Multiple system involvement, কানে ব্যথা স্যার, গলা ব্যথা করছে, পেটে ব্যথা করছে, একটু বুকে ব্যথা করছে. So multiple system are involved. That means there is no focal investigation or focal problem is not there. But suppose a patient coming with a gland enlargement here, only gland is enlarging, so we can suspect the patient may suffering from bacterial pump. Whatever it may be, you have to go for the complete blood count And this complete blood count means everything will come out, like the hemoglobin level, like the neutrophil level, total count of WBC level. We come day by day increasing in your class when I will continue my lecture in blood, the total count of WBC, what is the total count of RBC is there. But still remember, most infections in this season is happening in your family by toder karo hocche, mostly viral. But still there is an exception also is there, because I am listening to a story. 2 days ago, 2 days before, a patient come who is vomiting, loose motion and chest pain. A child having 8 years old, কি বললাম, vomiting, fever, chest pain, right sided chest pain, and also loose motions. So I thought this is mostly a viral, but the problem is that whenever a baby or any adult is complaining a chest pain specifically, so you have to detect some pathology must be there. That's why he or she is telling such problems with chest pain, and that is a huge pain. The child was crying, coming to my chamber with crying, a huge crying at that point. Normally this is the viral, but I do some X-ray, simple X-ray. I order the blood. I still waiting for their report. But in X-ray, lab called me immediately that the patient having huge consolidation, that means pneumonia in right upper lobe. That is important to be a good clinician. তাহলে মেডিসিনে কিন্তু 1 + 1 = 2 হবে না। So for that reason, these classes are important to you. How to get knowledge and how to use your knowledge in your practical life from now. এখন তোরা পুরোপুরি যে জেনে যাচ্ছিস জানবি তা নয়, but these classes will helpful to know that how you can reach a proper diagnosis. Right? Now, so complete blood count or count high asbe. Now I am confirmed because it is a consolidation, that means some bacterial infection is there. So neutrophil count will be high. So I immediately started antibiotic. Yesterday the parents and child had come to revisit, and I saw that the changes of sound is there. That is a different entity, is not interesting for you, but still I want to make this class to be interesting for the classes, because you are going to be a doctor, not only theoretical knowledge. No, they are not going to be a BSc, MSc. It's not. You will deal in future as a, you will deal patients. You have to save life. In our Santiniketan Medical College, is a faculties are very professional and good, and so gather knowledge day by day, and always try to correlate with each of them. Each of them correlations must be there. এখন থেকে অভ্যাস করতে আরম্ভ করলে অসুবিধা হবে না। So CBC and CRP, I am telling repeatedly, that's very important to know about the blood, why you are dealing with the blood. So now you can get some interest by reading your book. যখন তোরা এরপরে বই পড়তে যাবি, তখন তোরা এবারে ভাবতে যাবি যে why we should learn those things. এমন না যে ওটা জেনে কি হবে, এটা জেনে কি হবে, আমার সায়েন্টিফিকেশন জেনে কি হবে। আমার সায়েন্টিফিকেশন জেনে এটা লাভ হলো যে that's causing the plasma term, the serum term, RBC, Buffy code, platelet, everything is coming. Right? Now, the important part of this, up to components, these are the components of blood. That means components of blood means RBC, WBC, neutrophil. In the WBC means leukocytes. Leuko means white. Cytes means cell. Remember always. Leuko means white and cyte means cell. Like white blood cell. Erythrocyte. Erythro means red. Cyte means cell. RBC, red blood cell. Platelet is a different thing. And serum is equal to plasma minus clotting factor. Don't confuse with serum and plasma. Sir, এটা serum, না এটা plasma, এরকম না। কোনো কনফিউশন নেই। এই ফর্মুলাটা জীবনে ভুলবি না, SPC একটা মাঝে মাইনাস আছে। ঠিক আছে? So in the components of the blood, there is a important components of constituent of the plasma, that is called plasma proteins. Why it is important? Again, same, why you have to learn this plasma protein. Number of plasma protein are there. Already we describe one plasma protein, that is C-reactive protein. দেখেছিস আমাদের ডে টু ডে লাইফে মেডিসিন বল বা ফিজিওলজি বল, each and every time we'll came across the plasma proteins. This is also C-reactive protein, plasma protein. When I write as a CRP level, CRP level will come, which is more less than 6 is a normal level. If it is more than 6 or anything else, that is when some infection is there, you can't detect whether the patient is suffering from fungal, bacterial, viral, parasitical by seeing only the CRP level. That is also definite test is there, that is called procalcitonin. I am not details about it because it is beyond scope of your class, but that can be detected in the lab. We do regularly, we do practice the procalcitonin level. Procalcitonin level is, if it is high, that means severely suffering from the bacterial infection must. So we have to see use some antibiotics and treat the patient by seeing the CRP level. যে আমি একটা অ্যান্টিবায়োটিক দিলাম, CRP level কমছে না। তো CRP is also diagnostic as well as prognostic. এটাকে বলা হয় প্রগনোসিস। কেন? ভালো হচ্ছে কিনা, ভালো হলো নাকি আমি দেখব। So it is a diagnostic, আমি ডায়াগনোসিস করতে দিকে যাচ্ছি, and again we have to deal with the prognostic, whether it is prognostically good or helpful or not. একটা patient is suffering from bacterial infection, like my patient is suffering from pneumonia, I have added some antibiotics, and now we have to check every alternate day the CRP, what is going on. Suppose CRP is 100 or 80 and it is not coming down, that means the antibiotic is not working at all. So you have to check about the blood culture, whether it is which antibiotic will actually active for that child or any adult patients. So each and every step is there is a bar. তোকে সেই বারটাকে ক্রস করতে হবে with your knowledge, and that knowledge will come from the first day of the beginning of your MBBS class. এমন নয় যে আমি 5 বছর পরে শিখে যাব, হবে না। So plasma proteins is very important to know. One of this is a C-reactive protein, but overall plasma protein is present in the plasma, and there are mostly 3 types. There are plasma proteins mostly in 3 types. We can divide into 4 types also, but to some extent in good books it is written as a 3 types. What are those? Any idea? Yes. Albumin, very good. Yes, very good. Albumin, then globulin, very good. Fibrinogen. হ্যাঁ, এবারে কোন কোন বইতে এটাকে প্রথম দিন বলেছে, বলবি কোনো আপত্তি নেই, বাট আমি জানবো কেন, আমি MBBS পড়তে এসেছি, আমার এসব অ্যালবুমিন গ্লোবুলিন এসব জেনে আমার কি লাভ। So now I'm coming to your problem. So suppose a patient comes to OPD, outdoor patient department, এই যে OPD হচ্ছে, হ্যাঁ। So these patients come with swelling, whole body swelling, and as an intern, after passing your MBBS, you will continue your internship, and you see the patient is only complaining of swelling and soft sorts of respiratory distress. Patient age দিয়ে দিলাম, suppose 8 years old child, and so respiratory distress is there, and there is some swelling of all, পা ফোলা, মুখ ফোলা, পেট ফোলা, ফুলে দিয়ে চলে এসেছে। So edema, edema means fluid is coming from circulation to the interstitial space, right? So normally what happens? This is a blood vessel, suppose this is a blood vessel, and these are the interstitial tissue, তাই তো, এগুলো তো টিস্যু আছে। These are the tissues are there. Normally we have to hold the water inside the capillaries in blood vessels. ধরে থাকতে হবে, ইউ হ্যাভ টু হোল্ড। আমার তো ইডি মানে, তোর তো ইডি মানে, কেন এটা হচ্ছে? So there is some pressures are there, hydrostatic pressure, osmotic pressure are there. I will not come to detail at that point because in general physiology you will came across, কেউ না কেউ তাদের পড়াবে। Whatever it may be, you came across to some extent Stirling law, Stirling force, বলে যাচ্ছি একটু বই দেখে খুঁজবি। Stirling force, hydrostatic pressure, osmotic. What do you mean by osmosis? What do you mean by osmosis? Diffusion, osmosis. When the two separate chamber with some semi-permeable membrane, হ্যাঁ বল, কমের দিকে যাবে। কমের থেকে যাবে বেশি না বেশি থেকে যাবে? সলিউট তো যাবে না। একদম ঠিক বলেছে, হাইয়ের থেকে লোয়ের দিকে যাবে। কিরকম জিনিসটা হবে? এরকম আছে, একটা আছে, ঠিক আছে, সাপোজ এখানেতে জল আছে আর কিছু সলিউটস আছে, আর এখানে জল আছে, গাদা সলিউটস আছে, তাই তো? দেখ আমি বোঝাতে পারছি নাকি, এগুলো খুব বেশি আমাদের অত ক্লিয়ার কাট অত নেই, প্র্যাকটিক্যালি যেটা বোঝাবার জন্য বলতে চাইছি যে দিস ইজ সেমি পারমেবল নম্বর, সলিউটস উইল নট কাম, সলভেন্ট উইল কাম আউট। তো এখান থেকে এটা কি হবে না, এটা এই দিকে আসবে, তাই তো? দিস ইজ অসমোসিস, এটা কিছু নেই। সো দিস মেইনটেইনড বাই এ অসমোটিক প্রেসার, that may be capillary osmotic pressure and also interstitial osmotic pressure. Similarly, that may be capillary hydrostatic pressure and also interstitial hydrostatic pressure. So these four pressures নিয়ে Stirling force হয়। ডিটেইলস গেলে আর এগোনো যাবে না। আমি কেন এটা বলতে চাইছি, because these are coming for two concept that hold। আমার তো রিডিমা নেই কেন, নর্মালি আমাদের প্লাজমা অঙ্কোটিক প্রেসার, rather colloidal oncotic pressure, we can say colloidal osmotic pressure also is normal, that is 25 millimeter of Hg, আমার তো সবারই আছে। Which one is responsible for that? Albumin. Albumin is responsible for this to hold this oncotic pressure, that is 25 millimeter of Hg. তাহলে আমার এখানে অ্যালবুমিনস আছে, তাই তো? Normally, due to the hydrostatic pressure of capillary, the water can come into the interstitial space, আসবে কিন্তু এখানে, but due to the property of this oncotic pressure, osmotic pressure, these will drag the water back into the capillary. That is why we are not suffering from any types of edema. The patient we described, that whole body swelling, that may be any reason, but we have to first think the patient, whether patient is any albumin problem or not. Patient may suffering from cardiological problem also, but whether patient is coming for facial swelling, especially periumbilical puffiness, facial puffiness, that means their tissue is there, and normally this happens due to the cause of kidney problems and as well as our congestive cardiac failure also. Renal, আমাদের আগে মুখ ফুলবে না পা ফুলবে এই নিয়ে তোরা পরে আবার জানবে, এই ডিটেলসে আমি গেলাম। বাট হোয়াট এভার ইউ আর ডিলিং উইথ এ হোল বডি সোয়েলিং অর ইডিমা, ফার্স্ট কাম মাইন্ড যে অ্যালবুমিনের কিছু ব্যাপার নেই তো আবার। এবার অ্যালবুমিনের যদি কোনো ব্যাপার থাকে, সেই অ্যালবুমিনের ব্যাপার আবার হিউজ। হয় তার ক্রনিক লিভার ডিজিজ আছে, কারণ অ্যালবুমিন কোত্থেকে তৈরি হয়? লিভার থেকে। তাহলে ক্রনিক লিভার ডিজিজ আছে, বা অ্যালবুমিন আমার রেনাল দিয়ে ফিল্ট্রেশন হবে না, নেগেটিভ চার্জ, গ্লোমেরুলার বেসমেন্ট মেমব্রেন, তোরা আস্তে আস্তে পড়বি, আমরাই পড়াবো। সো ফিল্ট্রেশন ছাঁকনি করে নেবে, ছাঁকনি হয় নেফ্রন, হ্যাঁ, তাহলে অ্যালবুমিন যদি বেরিয়ে যায় দ্যাট ইজ কল অ্যালবুমিনুরিয়া, ইউরিন দিয়ে হাইপার অ্যালবুমিন বেরিয়ে যাচ্ছে। সো ইফ ইট ইজ অ্যালবুমিনুরিয়া দেন অ্যালবুমিন বেরিয়ে যাচ্ছে, ইফ ইট ইজ লেস এটা বেরিয়ে যাচ্ছে, এটা বেরিয়ে যাচ্ছে, এটা বেরিয়ে যাচ্ছে। How they can hold that pressure 25 millimeter Ag to hold the water in the capillary? Then water will be shifted to the interstitial tissue, presentation as a facial puffiness, pedal edema, everything is there. বোঝা গেছে? এটা খুব সহজ করে যেটুকু বোঝাবার তোকে বললাম। Now albumin is released from or synthesized from liver. গ্লোবিউলিন কোথা থেকে বেরোয়? হ্যাঁ, হইল না, কাছাকাছি যাচ্ছিস বলতে বলতে, হ্যাঁ, গামাটা আমরা পড়েছে। সো গ্লোবিউলিনস আর ডিফারেন্ট টাইপস। কি কি রকমের? আলফা, বিটা, আলফা বিটা গামা। আমি তাহলে লিখি alpha, beta, gamma. Very important for your theory also. Alpha, beta, gamma. তাই তো? Anyone আর কেউ কিছু বলবি? এটা ছাড়া? বল। Alpha 1, যা বলবি সব লিখবো। Alpha 1, আর কিছু বল? হইবে না, মজা নয়। ও পড়ে এসছে বলে বলছে। মজা হবে না। আলফা 1, আলফা 2, বিটা 1, বিটা 2 হইবে না। বিটা 1, বিটা 2 যদি কোনো জার্নালে বা থাকে আমাকে দেখাস, আমি জানি না, হতে পারে, দ্যাট মে বি। মেডিকেল সায়েন্সে জেনে গেছি এটা হয় না, আমি কালকেও জেনেছি, আজকেও জানবো, রেগুলার আমরা জানি। বিলিভ মি, আমরা সবকিছু জানি না। ডে টু ডে প্র্যাকটিস করতে করতে আমরা ডেলি শিখি, পেশেন্টের কাছ থেকে শিখি, তোরাও তাই ডেলি আমাদের কাছ থেকে শিখবি, এখন আমাদের কাছ থেকে শিখবি, ভবিষ্যতের কাছ থেকে শিখবি, বন্ধুদের কাছ থেকে বন্ধু অনেক কিছু শিখবি। তো শেখার কোনো শেষ নেই। সো আলফা 1, আলফা 2, বিটার কোনো ভাগ আছে নাকি আমার জানা নেই, প্রবাবলি আমার যেটুকু মনে আছে নেই। গামাতেও সেরকম কিছু নেই। অনলি গামা ইজ এক্সেপশন, দ্যাট ইজ কাম ফ্রম দ্য বি লিম্ফোসাইট অর প্লাজমা সেলস। ইট ইজ কল্ড ইমিউনোগ্লোবিন। ইমিউনোগ্লোবিন মিন্স কি কি টাইপের ইমিউনোগ্লোবিন? IgG, IgA, IgM, তারপর IgD, IgE. So IgG, IgA, IgM, IgD and IgE. Each and every function is different. IgG mainly seen in the chronic patients. Suppose you came across with the dealing as a jaundice. তোদের কারো না কারো ছোটবেলাতে jaundice হয়েছে, that is mostly viral hepatitis. So autoimmune hepatitis may presentation of this IgG level high. দরকার নেই। A means secretory, এই যে আমাদের intestinal, gut, immunity, IgA দেবে। Some sorts of our যেখানে আমাদের মিউকাস আছে, বেসিক্যালি মিউকাস, হ্যাঁ, সেইখানটা আমাদের IgA ইমিউনিটি দেবে। লাংসের ওখানে IgM মানে হচ্ছে অ্যাকুট। সেই জন্য দেখবি ডেঙ্গু IgM, এলাইজা মেথড, ডেঙ্গু হচ্ছিল কলকাতাতে, কি টেস্ট করছিল? ডেঙ্গু IgM, অ্যান্টিবডি দেখছে, ডেঙ্গু ঢুকেছে। আফটার 5 days they are forming antibody, and that antibody is type of IgM, that is acute, not chronic. Chronic is IgG and IgM is acute, and IgD is not fully known. IgE for allergic, anaphylactic reactions, any types of allergic tendency, its IgE level is high. Suppose you patient is suffering from asthma, suffering from some sorts of allergy rhinitis, in that patient's the IgE level will be high. আমরা পাই 1000 এর উপর, যেটা less than 200 সেটা 1000, 3000 এরকম থাকে। So these things are very much important for your practical life. That's gamma. Gamma is different from synthesis from the plasma cells or B lymphocyte. B lymphocytes এর পরে plasma cells। বিটা কিছু নেই, যে বলছিস আছে সে দেখাবি, কোনো অসুবিধা নেই। বিটা তাহলে কি কি হবে? এক্সাম্পল কি? বিটার মধ্যে এক্সাম্পল হচ্ছে কতগুলো নাম বলি, অনেক কিছু আছে, আমারও অত কিছু মনে নেই এখন। That is the transferrin. That is a transferrin. Ceruloplasmin is a plasma protein, no doubt at all. Ceruloplasmin is probably it is alpha 2. তাও তোরা দেখবি যখন বলে ফেলেছিস তখন দেখবি ট্রান্সফেরিন হিমোপেক্সিন তারপর এই যে সি রিঅ্যাক্টিভ প্রোটিন দেখেছিলাম সি রিঅ্যাক্টিভ প্রোটিন ইট ইজ এ বিটা টাইপ সি রিঅ্যাক্টিভ প্রোটিন and complement system এর দুটো পার্ট that is C3 and C4 component C3 এগুলো সব পড়া হবে পরে C3 and C4 component of complement system. Complement system is a different system. আমাদের ইমিউনিটির একটা সিস্টেম ইমিউনিটি যেমন আমাদের innate immunity acquired immunity you will came across day by day and so is the complement system also there. So transferrin vaccine এইটুকুই মনে and CRP and C3 C4. Now alpha 1 দুই একটা নাম কেউ বল আলফা 1 এর মধ্যে হচ্ছে থাইরক্সিন বাইন্ডিং গ্লোবিউলিন ওই যে বলেছিলাম বাইন্ডিং করতে হেল্প করে প্লাজমা প্রোটিনের কাজ কি কি ইচ এন্ড এভরি প্লাজমা প্রোটিন হ্যাজ ডিফারেন্ট ফাংশন কেউ একটা বললো না যে সেলুলোপ্লাজমিন ফাংশন কি সেলুলোপ্লাজমিন ক্যারিজ কপার এন্ড ট্রান্সফর্ম ফেরাস টু ফেরিক সেলুলোপ্লাজমিন ইজ এ প্লাজমা প্রোটিন and ceruloplasmin level is seen also in case of Wilson's disease. Wilson's disease আমাদের সেরোটোনিন তৈরি হয় না that's why copper deposition in liver and that may presentation is a jaundice আমি দেখেছি patient Wilson's disease is not so uncommon so this is the ceruloplasmin level so ceruloplasmin is a type of alpha 2 alpha 1 খুব মনে রাখার যে বিশাল একটা দরকার আছে জানা দুই একটা করে নাম তোদের কিন্তু মনে রাখতে হবে at least up to this কি শিখলি প্লাজমা প্রোটিনটা একবার একটু চালিয়ে নেওয়া যাক plasma protein means is present in the plasma and there are different types of plasma proteins are there plasma proteins mainly in case of 3 to 4 types that is the albumin that is the globulin that is a fibrinogen or you may say that there's a as a prothrombin basically prothrombinটা একটা বিটাতে অনেক সময় বলে দেয় বিটা টাইপ that's why it is called 3 types anyway so albumin is very important এগুলোর মধ্যে albumin is most important because this maintains our plasma oncotic pressure that is 25 millimeter of Hg mercury okay so this holds the water into the capillaries or into the our system circulatory system if it is fails যে আমার কমে গেছে কিভাবে কমতে পারে না আমার যেখান থেকে তৈরি হচ্ছিল কোথা থেকে তৈরি হচ্ছিল লিভার একটা chronic disease suppose example is the cirrhosis of liver suppose is a chronic liver disease cirrhosis of liver in that case albumin may be reduced and so it can't hold the water inside the capillaries and so the water spills into the interstitial tissue causing কি পাবো edema ক্লিয়ার হয়েছে এটা না গ্লোবুলিনের ফাংশন হচ্ছে নানান কাজ তার গ্লোবুলিন আমাদের অনেক রকম টাইপের হলো তাহলে এক এক রকম কাজ আমি কোনো ফিক্স কাজ বলতে পারি না and one thing you remember that not only the plasma oncotic pressure is maintained by the albumin globulin also may hold or help to hold the water inside the capillary 80% of holding is done by the albumin and rest of is done by the 20% always remember এর জিজ্ঞেস করতে পারে ভাইবাতে একটু ভালো বললে আমরা জিজ্ঞেস করি তারা কি শুধু অ্যালবুমিনই এটা করে নো অ্যালবুমিনই জিজ্ঞেস করে তা না গ্লোবুলিন এটাতে হেল্প করে সো is a globulin is a alpha 1 alpha আমি উপরে লিখে দিচ্ছি alpha 2 it is a alpha 1 and alpha 2 it is alpha 1 and alpha 2 alpha 1 example is thyroxine binding protein write down thyroxine binding protein that means thyroxine is a hormone is a thyroid hormone that is a T3 T4 hormone thyroxine is a T4 basically and T3 is called triiodothyroidine এটা এর আগে ভুল হয় এটা T4 means thyroxine T3 means not thyroxine and nowadays we if you suspect the patient is suffering from any hypothyroidism মানে level কম then we write only the free T4 and TSH means thyroid stimulating hormone separate thing জাস্ট এলো তাই বললাম। সো নাও দিস ইজ থাইরক্সিন বাইন্ডিং প্রোটিন অ্যান্ড এনি আদার নাম বল। ভেরি গুড, ট্রান্স থাইকর্টিন, এটাকে বলা হয় তোর এটাকে বলা হয় ট্রান্সকোটিন। Trans-cotin is corticosteroid binding protein. It is also alpha 1. And then one important thing, don't forget, that is the lipoproteins. Now one lipoprotein, that is HDL. এইখানে দুটো কথা বলে তোদের ছাড়বো এটা। সেটা হচ্ছে এই আলফা 1 এর মধ্যে আছে আমাদের আলফা 1 HDL বা লিপোপ্রোটিন, এটাকে বলে লিপোপ্রোটিন, হাই ডেনসিটি লিপোপ্রোটিন। আরেকটা জানিস যে তোরা LDL আছে। Now which one is good and why? Which one is good? HDL is good. Why? এটা তোদের রিলেটিভেরও রিপোর্ট তোরা এবার পাবি, দেখবি দেখ তো রিপোর্টটা কি আছে, লিপিড প্রোফাইল। সো ইউ হ্যাভ টু কনসেপ্ট high density lipoprotein means it transfer the fat from periphery to the liver. But LDL it from liver to the periphery. সেটা আমাকে ক্ষতিকারক, এটা আমার পেটে দিয়ে যাচ্ছে, আমি জমছে না। হ্যাঁ, so these are the bad, the LDL, and LDL is a part of কি বিটা টাইপের আছে তোর LDL, এইখানে আছে। বোঝা যাচ্ছে? So good cholesterol means HDL. Bad cholesterol is a LDL, because HDL it transforms fat periphery to the liver, but in case of LDL it liver to the periphery, যেটা খারাপ, সেটা আমি হতে দেবো না। এই জন্য বলা হয় যদি তোমার কোনো কোলেস্টেরল বেশি থাকে, you can eat eggs, large numbers of HDL is present. কনসেপ্ট অনেকের রং আছে, ঠিক আছে? HDL বলে যে ডিম খেয়ে না কোলেস্টেরল আছে, খাবো না, ভুল কথা, wrong. HDL level is high, so he or she can take, এমন নয় যে সে দিনে 4টে করে খাবে, বাট ইউ ক্যান টেক ইজ এ রেগুলার বেসিসে একটা করে খেতেই পারে, কোনো অসুবিধা নেই। সো দিস পার্টস ইজ অলমোস্ট ফিনিশিং। সো আলফা 1 and আলফা 2 যদি ভুলেও যাও তাহলেও কিছু এসে যাবে। এগুলো হচ্ছে অনার্স মার্কসের জন্য, আলফা 1, আলফা 2, beta and that. And celluloplasmin is important, as a short note it comes, as celluloplasmin acts in two ways, that is carries the copper, and also it transforms the ferrous to ferric Fe+2 to Fe+3, and so the function of plasma protein as a whole is not a straight cut. কখনো স্ট্রেট কাট হবে না। যখন কোনো ফাংশন অফ প্লাজমা প্রোটিন, এটা এক্সামে দেয়, প্লাজমা প্রোটিন বলবে, তখন টাইপস গুলো ভেবে ভেবে অ্যান্সার দিতে হবে। যেমন আমি অ্যালবুমিন ভাববো, আমি বলবো এটা মেনটেন্স দিস অঙ্কোটিক প্রেশার টু প্রিভেনশন অর হোল্ডিং দ্য ওয়াটার অ্যান্ড দিস ওয়ে। না, হোয়াট আর দ্য লেভেলস? লেভেলস কি আছে? অ্যালবুমিনের লেভেল কি থাকে? নর্মাল লেভেল কত অ্যালবুমিন, তবে তো বুঝবি যেটা কম বেশি। লেভেল কি? নর্মাল লেভেল কি? অ্যালবুমিনস নর্মাল লেভেল is 3.5 to 5 gram per DL. Albumin is 3.5 to 5 gram per DL, মানে 100 ml, 100 liter, and is a globulin. কত আছে এটা? 2.5 to 3.5 again gram per DL. Fibrinogen কত আছে? 200 to 400 mg per DL. এটা কিন্তু mg, not gram. So দুটো গ্রাম হলো, একটা mg হয়ে গেছে। So albumin if it is less than 3 then will come as a edema. তাহলে এটা জানা উচিত। The normal level is 3.5 to 5 gram DL. হ্যাঁ, আমার হাতে লাগাটা খারাপ, ঠিক আছে, you have to bear this. A globulin is a 2.5 to 3.5 gram per DL, and fibrinogen is 200 to 400 mg per DL. So if it is less than 3, then will come as a symptom. And if AG ratio is another important point. AG ratio, A is to G ratio is 2:1. That is also altered during the chronic liver disease or any types of acute viral hepatitis, primary biliary cirrhosis, etc., etc. Lots of diseases are there. do not want to exhaust your brain in the first class, তাহলে বলবি বাবা কি সব পড়াচ্ছে আমি কিছু বুঝতে পারছি না পাড়াই। So whatever it may be, always try to relate with the clinical scenarios why we should know the plasma proteins, because plasma protein এর মধ্যে অ্যালবুমিন আছে, তাহলে that may be edema cause গুলো আমার হবে, এডিমার মধ্যে লিভার ডিজিজ চলে গেল, কিডনি ডিজিজ চলে গেল, all coming. Now there is it is a globulin. Globulin acts is different way in different tests. যেমন ট্যাসফেরিন আয়রন ক্যারি করে, যেমন সেলুলার বায়োলজি কপার ক্যারি, তাহলে ক্যারি করতে হেল্প করছে। সিড অ্যাক্টিভ প্রোটিন আমার ইনফেকশনকে ইয়ে করছে, থাইরক্সিড বাইন্ডিং প্রোটিন থাইরক্সিড ক্যারি করছে। সো দিস প্লাজমা প্রোটিন ইজ ইম্পর্টেন্ট টু ক্যারি সাম হরমোনস, ক্যারি সাম ড্রাগ অলসো, যে যাকে আমার ফ্রি, দ্যাটস ওয়াই দিস ফ্রি ড্রাগ ইজ মাচ মোর ডেঞ্জারাস দেন দ্য বাউন্ড ফর্ম। If any hormone level is less, then the bound form goes to the free level. So আগে bound করে থাকবে, then it come if need, our body যদি মনে করে যে হ্যাঁ আমার হরমোনটা কমে গেছে, then albumin will release, যা বাবা তুই কাজটা করে আয়, কারণ তার আগে আমার যাওয়ার দরকার নেই। To some extent lipids are not soluble, it is insoluble in water. To make it soluble, এই যে গ্লোবুলিন ধরলো, লিপোপ্রোটিন গ্লোবুলিন ধরছে আমার, HDL কে ধরছে, LD কে ধরছে, তবেই তো ব্লাডের মধ্যে আমার চলাফেরা করছে, and that's why we are getting the HDL LDL level in the blood. তাহলে আমরা কি লিখি? লিপিড প্রোফাইল, সিরাম লিপিড প্রোফাইল, লিপিড প্রোফাইলের মধ্যে আমার কি করবে? কোলেস্টেরল পড়বে, ট্রাইগ্লিসারাইড পড়বে। সো দিস সলিবল না, তা সলিবল নয়, তাহলে তো হয়ে যেত, প্রেসিপিটেট করতে থাকতো না, কেন থাকছে? ডিউ টু দ্য বাইন্ডিং উইথ সাম প্লাজমা প্রোটিন। So plasma protein is binding capacity is there, transferring capacity is there. Also, it helps the infection defense mechanism also is there. Gamma means immunoglobulin. Simple IG is a immunoglobulin. Immunoglobulin structure is there. I will come across step by step in case of immunity chapter. But this will, as a whole, is a plasma proteins. বোঝা গেল? যদি বলে প্লাজমা প্রোটিনের ফাংশন লিখতে পারবি 5 মার্কের মতো, আমি বলছি না 10 নম্বর এলে এমন একটা যেন নলেজ নিয়ে যা, ইচ ক্লাসে ইউ হ্যাভ টু রাইট অ্যাট লিস্ট টু গেট 5 to 6 মার্কস, দ্যাটস এনাফ। আর যদি কিছু মনে থাকে তাহলে তুমি লিখলে আরো। এখানে অনেক তথ্য আমি দিয়ে রেখেছি, সব তথ্য যে তোমাকে মনে রাখতে হবে তা নয়, বাট বেসিক যেন ভুল না হয়। বেসিকের মধ্যে কি কি গেল? সিরাম প্লাজমা ডিফারেন্স আলাদা, নাম্বার 1। Number 2, why we should know that the CBC or the complete blood count কেন জানতে হবে, ভাইরাল ব্যাকটেরিয়াল প্যাটার্ন কি জিনিস, কেন এগুলো আমি জানবো, কেন লিখবো। C reactive protein, it is a type of plasma protein and it detects, it helps whether any inflammation in your body is whether is present or not. A patient is suffering from some pain, sorts of pain and lots of fever is there, but CRP level is to some extent level, that means 8 to 7 হয়েছে, চলো ঠিক আছে আরো কিছুদিন দেখব। The prognostically is important, whether it is increasing or decreasing or not. If we use in the antibiotics, also follow-up cases we do the CRP level to see the whether the antibiotic is working or not. Is the indirect prognostic value of the CRP. So these things will come in the clinical scenario, and this plasma protein is very much important for your albumin, globulin and fibrinogen. One of the last is the fibrinogen. What is the function of the fibrinogen? Fibrinogen function is the clotting factors. That means it forms prothrombin to thrombin and thrombin fibrinogen to fibrin. Fibrin is a fibrin mesh. I am coming later day by day in case of clotting factors. What is the how clotting is formed? Details. I mean, this if they allow me to give some lectures and see I give details of how this clotting factor is happening with the clinical scenarios. Some congenital diseases are also there. So fibrinogen is for the our clotting factors or coagulation factors is there in plasma protein. So plasma protein function একটা coagulation cascade এর কথা উল্লেখ করতে হবে। Always remember the types and then their functions. And alpha 1 an example, alpha 2 একটা দুটো, beta and then the gamma. Gamma means these are the gamma. This is IgG, IgA, IgM, IgD and IgG. Everything is different and different functions are there. বোঝা গেছে? বুঝেছিস? 50 to 60% বুঝলেও অনেক। বল। ডিফারেন্স হচ্ছে বিভিন্ন ডিজিজে, ক্রনিক মানে হচ্ছে অনেকদিন ধরে জিনিসটা হচ্ছে, অনেক দিন ধরে কোন একটা পেশেন্টের মনে করো আমরা বলছি যে ক্রনিক ডায়রিয়া। ক্রনিক ডায়রিয়া মানে ক্রনিক কাফ, এখন তো অনেকে ক্রনিক কাফ হচ্ছে, সে হয়তো 1 মাস 2 মাস হয়ে গেছে। এক একটা ডিজিজের কোন ক্রনিক ডিজিজ বলে 6 সপ্তাহের উপর হলে তাকে বলে ক্রনিক, 6 সপ্তাহের নিচে হলে তাকে বলে অ্যাকুট। মনে করো অ্যালার্জিক একটা রিঅ্যাকশন হচ্ছে তার, হ্যাঁ, একটা আর্টিকেরিয়া, ক্রনিক আর্টিকেরিয়া, অ্যাকুট আর্টিকেরিয়া। Articaria means some rashes are there. If it is present more than 6 weeks, that means it is a chronic. Chronic means অনেক দিন ধরে, acute means এখনই হয়েছে, just এই কিছুদিন আগে হলো, হ্যাঁ, that is the acute. And there is a middle term is that is the subacute. Subacute মানে আরো নিচে আছে, হ্যাঁ, appendicitis এর আমরা বলি অনেক সময় is the subacute phase, subacute appendicitis, so এখন অপারেশন লাগবে না, আমি একটু অ্যান্টিবায়োটিক দিই তাও ভালো হয়ে যাবে। These schemes are subacute. Acute যায়নি, subacute, acute and chronic. And chronic disease always is so that G level is coming and acute phase is coming as the IgM level is coming. So whenever you are doing the dengue, typhoid, Ig level, immunoglobulin level, when antibody তৈরি হয়ে গেছে, একটা salmonella typhi is causing the typhoid fever and this forming the antibody in our, যা কিছু ঢুকতে তার অ্যান্টিবডি কিন্তু তৈরি হচ্ছে আমাদের। How it is coming, I will give lectures day by day. And this antibody is formation when it is the acute phase, that is IgM. Always we are writing that typhi IgM. That means I want to know that the typhoid antibody is acute condition, IgM level টা বাড়লো কি বাড়লো না। যদি আমার IgM level থাকে দিস ডাইরেক্ট ইস হয়ে গেল। ডেঙ্গু IgM, আমি IgG নিয়ে আমি don't bother about that. কোভিড অলসো, কোভিডেও আমরা কিন্তু IgM এর অ্যান্টিবডিটা বেশি পাই, IgG টা আমরা ব্যাকটা কোশ্চেন করে দিই যে আমার আগে কোভিড হয়েছিল। আমার যদি আগে কোভিড হয়ে থাকে, আমার যেমন হয়ে আছে, আমার IgG তুই অলওয়েজ পাবি, আস্তে আস্তে গ্র্যাজুয়ালি কমে যাবে অবশ্য। বাট IgG লেভেল অনেক 3 মাস আগে আমার কোভিড হয়েছিল, আমি জানি না যে হয়েছিল নাকি, আমি জানি না, টেস্ট করাইনি, বাট সিমটম ওয়াজ দেয়ার। অনেক পেশেন্ট এরকম আছে যাদের আমার কাছে পরে এসছে যে আমার এরকম বাচ্চা হচ্ছে, পা ফুলে যাচ্ছে, সেটা সিমটম আমাদের দেখতে হয় যে বাচ্চাটার আগে কোভিড হয়েছিল না। সো ইউ হ্যাভ টু ডিটেক্ট দ্য IgG অফ দ্য কোভিড, IgG কোভিড, আমরা লিখেই দেবো যে IgG কোভিড করো। ইফ ইট ইজ পজিটিভ দ্যাট মিনস ওর আগে কোভিড হয়েছিল উইদিন 3 মান্থসের মধ্যে। নাও দ্য কমপ্লিকেশনস অফ দ্যাট রিঅ্যাকশন ইজ কামিং দ্যাট ওয়ে। So this way IgG is chronic, IgM is acute. Okay, simple. Thank you very much. Dismiss my class. Yes.
Practical, biochemistry practical. So mostly in your higher secondary, same your chemistry laboratory was there. In chemistry laboratory, what you learn? Some acid, some alkali, and buffer. All these things already you have known already. So here regarding your biochemistry practical, last already introduction class is over. Okay. So here what we will deal with? Why we will do biochemistry practical? We will deal with what? Your what test we will do? And that test, it may be from blood, it may be urine, it may be gastric juice, it may be CSF, it may be sweat. Okay, so regarding blood, so what is the syllabus in practical? So syllabus, all of you download from NNC website. Okay, website. UG curriculum volume 1, all of you download. Okay, UG curriculum volume 1. Okay, all of you download. From there you see what are the lecture class, what are the SCT, seminar, all these things written. Okay, one is your practical part is there, everything, anatomy, physiology, biology, everything is there. Lecture classes and what are the practical classes. Okay, regarding practical classes, your describe commonly used laboratory apparatus and equipments, good safety laboratory practice and waste disposal. Describe the preparation of buffers and estimation of pH. Describe chemical components of normal urine. Okay, and here one thing you have to see, See here, syllabus is there. Here all these things written there. Lecture, small group discussion, okay, and perform urine analysis to estimate and determine normal and abnormal constituents. Understand? What is written here? DOAP written. DOAP. Have you seen? Yes. DOAP. What is DOAP? DOAP. That means D for demonstration. Okay. O for observation. A for assist. P perform. Okay. D for demonstration, O for observe, A for assist, P for perform. That means whatever practical will be there, we have to do first demonstrate. Okay. Then after demonstration, what is there? Observation. That means what? How we are demonstrating. That is observed there. Okay. Then after that, next is go for using assist for practical. Next, perform. So higher level of knowledge is perform. That means skill based. Once you will perform the test, that means you are skilled. So skilled, that means according to IMG, so Indian medical graduate or you, all of you, will be a skilled doctor. Okay, skilled. So sample, suppose you take what you take first is your blood sample. Okay. From how you collect the blood sample? From where you collect blood? From where you collect? Blood sample? Tip of the finger. That is what kind of collection or what kind of blood that is? That is correct blood. Not finger also can take. What is that? That is? That is capillary blood. Huh? One is here. That is correct list. Or capillary blood. Okay. Another? Any other? Any other? Venous blood. Mostly the most of the case will do from the venous blood. Okay, venous blood. From which vein will take? Which vein? Now on the started. Okay. Which vein will take? Anticubital vein. Okay. Started blood how you collect? Which artery? Or? Okay, so venous blood and arterial blood. One thing I forgot. This capillary blood you will take from here. Here. Okay. For adult, suppose the newborn baby, from where you take? Newborn baby. Grade 2 is there. From grade 2, apply or heat aids. Okay, heat aids the baby. This is here regarding blood collection. Now, any other test what will do? Other test? Urine also take.
सर लास्ट डे को रिपीट करो
Huh?
लास्ट डे যেটা বললেন না रिपीट टू
Grade 2 or aids of healing. Healing aids. Okay, after collection blood, what will do? Will go for processing test. For that, we require anticoagulants. Any anticoagulants you can you name? Very good. Heparin. Any other? Eh? Any other? One is heparin. Very good. Any other? Then any other? Sodium chloride, ETTA, oxalates, and sodium chloride mostly we are using for blood sugar estimation. Okay, blood sugar estimation mostly sodium chloride. Why importance is there? That sodium chloride. What is glycolysis? Glycolysis means breakdown of glucose. Breakdown, very good. Breakdown of glucose. So end product, what will be the end product? Yes, that is pyruvate or lactate. Okay, pyruvate or lactate. So what will happen? Sodium chloride, what will happen? That one enzyme is there, enolase. Okay, that sodium chloride it will inhibit the glycolytic pathway. So accurate value you can observe. Regarding anticoagulants, one class will take later. Okay, separate anticoagulants. Next, here you can take urine sample. Okay, urine sample you can take for examination. How much and when? When you will take? Most people. For blood sample collection, also preferable time is there. Blood sample collection, preferable time is there. What time should be there? So for different tests, different tests. Suppose example will take blood sugar estimation. Fasting blood sugar. Suppose fasting. When will take? Suppose. For that we want. How will take? Empty stomach should be there. Up to what time? Mostly it should be 8 to 12 hours fasting should be there. Okay, 8 to 12 hours fasting. And And suppose BPBS,
After 2 hours.
2 hours.
what is BPBS code for BPBS?
Postprandial.
Postprandial. Postprandial procedure. MBBS code for fasting procedure. When you take this?
After 3 hours.
After 2 hours. When suppose morning we get fasting sample. Okay, 8 to 12 hours fasting was there. Morning 8 to 12 get sample. Okay, after given sample, he came and took breakfast. 8:15 started breakfast. Completed 8:30. Breakfast complete 8:30. 8:30. Okay. Then at what time you take this?
10:30.
When?
10:30.
Huh?
10:30.
Started 8:15.
10:30.
Just started. Salinization once that. From there metabolism started. So 30% eating time is good. 30% carbohydrate will be easily digested in mouth, through mouth. So when you started first salinization, from there it will come. That will take immediately. You take carbohydrate. RBS, what is the RBS?
Random blood sugar.
Random blood.
Random blood sugar. When you take at any time. Now regarding the urine collection, urine sample collection, morning urine sample should be taken. Okay. And urine test what we do? What can regarding blood sample, what test you can do? He can test for the blood name. Tell various test names. What you know? Very good. HbA1c. Any other? That is not direct biochemistry. You can do, but you will do. So this is not our part. Okay, next. Any other? Yeah. Okay, next. Any other test? Tell, so many test name is there. Creativity, very good. Urea, yes. Any? Something tell me. Sodium potassium chloride. That is electrolytes. Yes, that is correct. Electrolytes. Next. Any other? Any other test? Remember? Yes. T3, T4, TSH. This is your total T3. Total T4. Next is the FT3, FT4. 3T3, 3T4. Okay, very good. This is the thyroid profile. Okay. Very good. Next.
Say LFT.
Yes. LFT. That is
Liver function.
liver function test. RFT. Yes. That is renal function test. Under LFT, what are the tests are there? Yes. Next. Real liver. substances regarding kidney? Urea, creatinine, sodium, potassium, yes, chloride also, bicarbonate also. Okay. And regarding LFT, albumin, where it is present? Is it in liver? Globulin, any other? OTPT, SGUT, SGPT, or also called as the STLT. SGPT, SGPT. Serum, glutamate, or the transaminase. Serum glutamate, pyruvate transaminase, also called as SGPT. HB? Yes. Yes. Antigen test. Any other? So many tests are there. Okay. So many enzymes are there. ALP, alkaline phosphatase. Different markers are there. Different markers are there. What is that? This is marker. This is PSA, prostate specific antigen. In case of prostate cancer. Okay, so many different markers. Another is CA125. And so many enzyme tests there. We can do so many enzymes. Then other tests. Very good. See, everything is coming from his mouth. That is very good. What is your name? Amylation ligase. Regarding the blood sample collection, this test we can do. Urine. Huh? Urinary, routine examination. So what test we can do? In urine also. Creatinine, urea, everything we can do. So test regarding the urine test, urine collection, test we can do. And here what we will do in urine, mostly important is there, that is normal constants of urine, you can see normal constants of urine and abnormal constants of urine. Okay, what are the normal constants? What are the abnormal constants? From your exemption point of view, abnormal constituents of urine is very important. Okay, we will do there. What will do there? Very important thing here.
Thank you.
Okay. What test will do there? will do BBC sugar, will do ketone bodies. Protein, yes, will do bile cells, bile pigments, blood. Okay, this test will do unknown person. Okay. Regarding your gastric juice, what test will do for what? Gastric juice? Huh? HCl, so we collect from rice cube. Okay. Then CSF, regarding CSF. CSF is the cerebrospinal. Same also can collect CSF. After CSF, how to collect CSF? Will that be also separate class is there? One class for CSF, cerebrospinal. How it is collected? What kind of composition? Okay. In which disease the level will be protein or sugar level will be increased and decreased. Everything will be read in details. In CSF also we can collect. How to collect that is very important. So lumbar puncture. CSF collection is very important. When the needle is here, the needle will take from the CSF sample by lumbar puncture between L3 and L4. Then what examination will do from CSF protein, okay, CSF sugar, CSF sodium, potassium, everything you can estimate. Next, any other test? Any other? Okay, mostly sweat also can do. So from sweat we can see how much chloride is there. Level of chloride. So you see, describe screening of urine for inborn errors and describe the use of paper chromatography. Paper chromatography also will do in practical principles of colorimetry, because for quantitative estimation we will see what we will say through colorimeter. What we will say? We will say color of the solution. What we will measure from color of the solution? Optical density. Optical 10 cm measure. So how that is principle and under upon which principle it acts under the which principle? Beer and Lambert law. That will read later. Estimation of serum creatine and creatine clearance. Serum creatine and creatine by the Japanese method, how they are doing, and principle, the normal rule, everything will read. Demonstrate estimation of serum total protein. Serum proteins, albumin and Ag ratio. Albumin, globulin ratio. What is the ratio? Yes, that way. Estimation of serum total cholesterol and HDL cholesterol. Estimation of triglyceride. Demonstrate estimation of calcium and phosphorus. Okay. Calcium, phosphorus, serum bilirubin, SGOT, SGPT, already told. Estimation of ALP, alkaline phosphatase. Describe and discuss the composition of CSF. And what are the common used equipments or techniques? That is your vapor chromatography, electrophoresis, PLC, electrolyte analysis by IAC, ABG analyzer, arterial blood gas analyzer. That will be later. ELISA, auto analyzer, quality control. Auto analyzer, one is semi-auto analyzer, another is fully auto analyzer. Here also semi-auto analyzer is there. In central lab you can see here fully auto analyzer. Principles of spectrophotometry. Spectrophotometry already last class we have seen. And very important is here, identify abnormal constituents of urine, interpret the findings and correlate with this pathological state. That is very important. Suppose we will see reducing sugar. How much sugar is in urine is there and in which disease it will increase, it will decrease. We have to correlate. Okay, these are your regarding practical. So, in demonstration copy, every class has practical step, bring your demonstration copy. Okay, from there, whatever, everything will write there, and whatever required from practical method, that only you will copy paste from here, demonstration copy to practical. Okay, then let it out and we will discuss. Last. Have you understood? What you understood? Tell. Yes, tell. No problem. Huh? Tell. What you understood? Regarding this class, what you understood?
Demonstration of blood.
Demonstration of?
Blood examination.
Yes, blood examination. From blood sample, what test can be done? That is one thing. What test you can do? Two tests. You can name two tests. Just name two tests.
Blood serum creatine.
Serum creatine, very good.
Fasting blood glucose.
Fasting blood. Sit down. What is your name?
Gali.
Huh?
A of the liver.
Air.
Of the liver.
Next. Tell me. Another two tests. Huh? You said here. Sit down. Very good. What is your name? Huh? Sauria. Next, last. Tell two test name. Huh? What? PP? BC. Not BC. PPBS. PPBS. Very good. What is full form of PPBS? You have written or not?
No.
Where is the demonstration copy? Not written PPBS.
No, previous written, sir, not full form.
Okay, what is that written? What written? What is full form of PPBS?
Not full form written, sir. I have written.
Why? That is your lacuna. Whatever I am telling, it is not understood. Same time you can ask. Okay, that is written postprandial blood sugar. Okay, postprandial blood sugar. Okay? Okay, sit down. Next.
Sir, LFT,
LFD, very good. What is the full form of LFD?
liver function test.
Liver function test. Under liver function test, what will you do?
Sir, bile pigments, cholesterol, albumin and globulin.
Okay, sit down. Your back. You tell. Two enzyme test. What enzymes you can do? Amylase. Very good. The you.
Protease test.
Huh?
Protease test.
Ah, you can do. Alkaline phosphatase. Yes. Okay, you can do. Tell any other test. Whatever they told. Regarding blood. Whatever they told. Yes. That is hydrogen. T46, F3, F4. This is the. You tell. Next, what else you do? I'm from blood. They told. Already told.
HBSG.
HB A1C. HbA1c, HbA2. Okay. Tell any other test left?
MBS, RBS.
MBS, RBS. Somebody told?
No, she said PPBS.
Yes, okay. Good. Next, Nandi, you tell.
Sir, sodium fluoride test for blood sugar estimation.
Sodium chloride, that is anticoagulation. That is anticoagulation. Other test what?
HbASG.
It is already he told. HbASG. Any other? Remember?
CA120.
SUT, Any other?
CA 120.
Yes. Yes.
So, what is full form of HBSG?
Prostate specific.
Specific antigen. Okay, sit down. Any other test?
Sir, all the electrolytes.
Or electrolytes? Calcium also can do? Yes, sir. Calcium also can do. Very important. Vitamins you can do. Vitamins also you can estimate. Yes. Hormones you can do. Prolactin, LH, FSH.
HCG also can be.
Yes, yes, you can do.
HPLC.
ACG you can do. FSH means full form. Very good. LH? Yes, you can do. Any other? Okay, little bit just replacement. What anticoagulants will utilize? Oxalates. And what are the restrictions are there, that also we will read later we will read. In which estimation you should not use oxalates? Example calcium. When you go for calcium, oxalates you can use. Like that, regarding anticoagulants, you take another class. Okay, thank you. Thank you to us. Any doubt? Any other doubt? Slowly. Okay, slowly we'll progress. Now we got overall idea. Overall idea. At this stage, we can forward with the biochemistry. Okay, out of that, all tests we can do. Regarding syllabus, whatever syllabus is there, we will do. Regarding syllabus, here only after normal concepts we read, unknown concepts we read, and all these things are there. Good, good tests are there. Okay, that's all. That's all. Okay, thank you. Thank you.
Okay, myself is Dr. Abhimita Roy. Okay, I have done masters in this specific subject. Today is your first introduction class. Okay, so today we'll be going about the general anatomy. Okay, so have you got any previous class? Secondary class 1, general anatomy? No one. So, what are the ideas you have? General structures, right? Our human body is made up of different type of connective tissues, right? What are they? What are the different type of connective tissue? Do you know? You are already passed class 2, right? Bone, cartilages, veins, blood, muscles, and tendons, ligaments. These are the connective tissue. So, for knowing the human body, for knowing the anatomy, at first structure you should know that is the bone. Okay. So, how many bones the human body consists of? 206. Very good. So, in the face or cranium, we are having how many bones? Cranium. Actually, skull. So you are confused. It is about 22. Okay, total 22. Now in the skull, see it is divided into. There are cranium. Okay, and there are facial bones. Right. So cranium means which is making the particular skull. Okay, the covering of the brain. So write it down. The human body consists of. Okay, so the human body consists of 206 bones. 206 bones. Among them, the skull consists of 22 bones, which is further divided into cranium, and the other bone present in the face, So here I will tell you some mnemonic, how to remember. Okay, so in the skull, see alphabetically letter H, head means H, right? So alphabetical letter H comes in which number? A, B, C, D, E, F, G, H. Total 8. So cranium or head consists of 8 bones. What are they? Write it down. Cranium or head consists of 8 bones. Among them, 4 are unpaired. Unpaired means single in number. 4 are unpaired and 2 are paired. So at first we will go for which are the unpaired bones. So if you see here in the skull, okay, so in front you could see this bone that is known as frontal. So frontal is 1, it is unpaired, frontal. Okay, so front means anterior. Then back. Back we have got another bone that is known as occipital, that is also unpaired. Okay, so these two bones we could visualize from outside. Okay, now if you just see the base of the skull, that means if you divide the cranium from the skull, internally you will see two other bones that is known as sphenoid and ethmoid. So these are also unpaired. First one is sphenoid and ethmoid. Okay, sphenoid and ethmoid. And which are then paired bone? Parietal and temporal. So if you see our air lobule, where it is situated, this bone is the temporal. Okay, and the bone which is making the wall, parietal means this is making the wall of the skull, both sides. So that is known as parietal. So these are the two paired bones. Okay, so total how many number? 8. Remember this one: frontal, occipital, sphenoid, ethmoid, 2 parietal and 2 temporal. These are bilateral. Okay, so this is about the skull. Next one is facial bone. So facial bone, how to remember? First one is you have to see about the nose. Okay, so nose, remember the letter 9. Okay, so total 9 bones are there. These are mnemonic. Please do not write on your exam, just to remember. Okay, 9 bones which are there. Could you name this nose? Huh? Yeah, you know the name, but how many number? Write it down. Nasal 2. There are 2 nasal bones. Which are nasal bone? This one, this notch, which is making the notch. Okay, that is known as nasal bone. There are 2 nasal bones. Then 2 lacrimal. Although lacrimal sounds like it is something of the eye, but it is a bone of nose. Okay, where it is found? If you see the orbital cavity, the inner margin, there is lacrimal bone. Okay, so 2 nasal, 2 lacrimal, then 2 palatine. Okay, then 2 inferior nasal concha. 2 inferior nasal, in plural you say concave. There are 2 inferior nasal concave. Okay, and one unpaired, that is known as vomer. VOMER, vomer. Okay, so in nose you could remember there are 9 bones. Okay, then next one is in the face. So face you remember 5. Okay. So which are the bones? 5 bones. Very good. 2 are maxilla. Okay. Then mandible how many? 1. Then another 2 bone. Zygomatic, which is making the smile. Okay. So cheekbone. So this is the zygomatic. So there is 2 maxilla or maxillae. Attendance. Okay. So there is 2 maxilla. Maxilla is actually making the upper jaw. Our teeth is having 2 jaw, upper jaw and lower jaw. So upper jaw is made by 2 maxilla and lower jaw is completely made by 1 mandible. Okay. So during development, mandible also 2 in number. Okay. Gradually it unite together. You could see in between there is one group. So that is known as symphysis menti. Okay, symphysis menti. Whenever you will get the symphysis term, that means it is a midline joint. Okay, symphysis pubis. Okay, then the symphysis menti. So these are the midline structure. Okay, so that is about the face, right? So how many bones? Next one is ear. So total 2 ear. And in the later year there are 3 numbers. So this thing you know, Malleus, Incus, Stapes, so total 6. Ear consists of 6 bones. Okay. Then one is hyoid, where it is situated in the neck. Remember, in the neck only one bone is there, that is hyoid. So what is the exceptional thing about hyoid? It has got no other bony attachment. Okay, it is suspended by different muscles. It is hanged by muscles, hair. Okay, it is protecting the front structure in the neck. So that is the hyoid. Okay, so how many bones? Just count it out.
29.
Including ear, you have to include all.
29.
Total 29. Okay, but we won't include ear bone. Okay, if you just exclude that, so you will get 22. Okay, so next will come. Next one is about. So you see, skull is over, right? Then there are two types of skeleton. One is axial skeleton, another one is appendicular skeleton. So write it down. Which are the axial skeleton? Axial skeleton means which are present in the trunk. Okay, trunk or body. So which are the axial skeleton? So first one is ribs. Okay, so first one is this bone is known as sternum. How many ribs are there? 12 pairs. Okay, you have to say 12 pairs of ribs. So that means 24. So 24 plus 1 sternum is 25. Okay, so in the chest you write there are total 25 bones, or rib cage there is 25 bones. 1 sternum and there is 12 pairs of ribs. So there are numerous classification in ribs also. We will go later on. Then which are the other axial skeleton? Vertebra. How many vertebrae are there? 6.
24, and 5.
Not 26. 33. Okay, remember 33. So you have to count all. Whether it fused or not, that doesn't matter. You have to count all. So how many cervical? 7 cervical. Then thoracic 12. Then lumbar 5. Then sacral 5. Coccyx 4. Okay, so total 33. Now if comparative anatomy, if you will ask, how many spinal nerves are there? Could you say?
Spinal nerves.
Cranial nerves are there. How many cranial nerves are there? Cranial nerve कितना है? Cranial nerve 12, right? So spinal nerve 31. How will count? So cervical is how many? This is 8. Okay, that's why I asked you. This is very common thing to confuse. Okay, first don't write it down. Just write it down. Keep one bracket. This is another topic. Okay. So how many cervical nerves? Cervical nerves are 8. Thoracic is 12. Then lumbar is 5. Sacral is again 5. Okay, and coccyx is 1. So total 31. Okay, so this is about the different spinal nerves. So how many vertebra? We are already there. Now we will come about the appendicular skeleton. Right? So in the appendicular skeleton, how many are there? You could see the upper lip and the lower lip. So in the upper limb, first thing you will have see that is known as shoulder girdle, right? So shoulder girdle consists of how many bones? Clavicle and scapula. Clavicle and scapula. So clavicle how many? Both sides 2 and scapula also 2 back side. Okay. Then in the upper limb, how many bones you could see? First one, humerus. That is known as shoulder joint. The head of the humerus along with the glenoid cavity of scapula, not clavicle. Remember. Okay. So that is making the shoulder joint. Then next one is next to what? Radius and ulna. Now the question is which side would be radius and which side would be ulna? Radius would be always towards the thumb. Okay, and ulna midline, medius. Okay. Then in the hand, how many bones?
8.
So which are the small bones, short bones, right? We will ask this one are the short bones. So how many short bones are there? 8. So these are known as carpal bones. So could you name them?
Scaphoid, lumen, triquetrum, visible,
Capital. Very good. So write it down. There are 8 carpal bones. First one is scaphoid. So how you remember scaphoid? It is looking like a board. Okay, board shaped bone. So that is scaphoid. Then lunate means looking like a half moon. Triquetral. Triquetral. Then pisiform. Okay. Then trapezoid, trapezium, capite, amen. So one thing in the Viva, examiner used to ask, tell me which is what, which bone. Okay. So at that time you have to identify at least scaphoid, which is scaphoid, which is lunate. This is very easy. Then pisiform. Pisiform is a small P-shaped bone. Okay, so pisiform also you could identify. So these are the 8 carpal bones. Then next one is phalanges. How many phalanges are there? 40. Right? So in the thumb we have 2. Rather than each finger has got 3 phalanges. The name of the phalanges is proximal, intermediate and distal. Proximal, intermediate and distal. So these are the phalanges. So that is all about the upper limb. Now coming to the pelvic girdle. So pelvic girdle, how many bones are there?
3.
So hip bone is consist of together. It is a type of irregular bone. It consists of 3 parts. Okay, first one is ilium, the fan shape. Okay, then which one is the ischium? Lower part. Okay, and pubis is the front part. Okay, so these are the 3 bones. So write it down. Hip bones, ilium, ischium, and pubis. Okay, ilium, ischium, and pubis. Okay. Then at the back, which bone is there? Which vertebra is there? That is sacrum. This you have to remember. It is making sacroiliac joint. Okay. Posteriorly there is sacrum and coccyx. Okay. So it is making the whole pelvic girdle. Okay. Then in the lower limb, which are the bone? The longest bone of our body is femur. So by the femur, length of the femur, you could identify the height of an individual. Okay. How much would be the height? Remember, height of the individual it is about 1/4 of femur. Okay. So next one is there is one bone in between that is known as kneecap, that is known as patella, and both sides medial and lateral. Medial side is thick bone that is known as tibia, and this side is lateral side. This is known as fibula. So these are the bone. Then at the feet, how many bones are there?
7 tarsal, 5
7 tarsal and 5 metatarsal and 14 canaries. Just like the hand, right? So how many tarsal? Name the tarsal.
Calculus, talus, tubercles, lateral, medial, inter.
Okay, well tried. First one is talus. First one is talus. Then which is making the heel, that is known as calcaneum. Okay. Then there is one tricky thing. There are two bones. Front. In the front row there is two bones. One is navicular, one is cuboid. Now which one is medial, which one is lateral? You remember navicular, N and M. Okay, N and M are proximal. So navicular are in the medial and cuboid on the lateral. Understood? So in the anterior row there would be navicular and cuboid. And again most proximal row there are three cuneiform. So which are they? Towards your body is medial. So medial, intermediate, lateral, cuneiform. Okay, so write it down. First one is talus, calcaneum, navicular, then cuboid, medial, intermediate, lateral, cuneiform. Three cuneiforms. Medial, intermediate, lateral, cuneiform. Okay, and then there would be 5 metatarsal. 5 metatarsal. And 14 phalanges. Okay, so now it would be the 206 bone would be on your tip of tongue, right? So this thing, this mnemonic you have to remember and you have to name the particular bone. Okay, so here is one thing we left about the classification of rib. How many types are there? So there are typical rib and atypical rib. How to know which one are the atypical? Typical means same characteristic. Same characteristic. Atypical would be first and second, 11 and 12. Okay, then true and false rib. True rib means which are directly connected to the sternum. So which are the directly connected? 3 to 7. Okay, and remaining are the false ribs. So these are the classification of ribs. There are numerous classification like vertebral, vertebrochondral. Okay, then vertebrosternal. These are the classification of ribs. Okay, now we will be coming about the bones. Okay, so general anatomy of bones. First one, what is your up to now how much you know? What is the general structure of bone? Bone is a type of connective tissue, right? That means it will have one covering that is known as periosteum, right? And there will be one intracellular matrix, right? And there will be some cells which are embedded on the matrix. So which is that thing is called? Which are the cells? How many type of cells? Do you know? Bones have how many type of cells? Actually 4 types. Okay. So write it down. The general anatomy of bone. So bone is a type of connective tissue. Okay. Bone is a type of connective tissue. Which is consist of, first one is there is one outer covering that is known as periosteum. Outer covering that is known as periosteum. Intracellular matrix. Now the question is, what are the structure present on the intracellular matrix? So could you say? Actually, it consists of collagen fibers. Okay, how many? About 40%. Okay, brain parenchyma or brain matrix is consist of 40% of collagen tissue. Okay, and there are two salt. What are they? Two minerals, calcium and phosphate. But remember, they are in the calcium hydroxyapatite group. Okay, they are present in the form of calcium hydroxyapatite. Okay, so that is one of the stone of the mixed stone of this calcium and phosphate. Okay, so these are the general structures. So bones are forming the rigid framework of the body. First one. Okay, you could have these notes. Then bones is highly vascular. Remember this one. It is highly vascular. Although it is a rigid structure, how many vessels do you know it's running? There are 4 type of vessels that run to the bone. If you see one bone very closely, you will find one foramen in between. That is known as nutrient foramen. So from that one artery is going and giving the blood supply. Okay. Then there would be periosteum. So there would be periosteal arteries. Then there would be juxta epiphyseal arteries and epiphyseal arteries. So it would be supplied by as well as 4 type of arteries. Okay. So it is highly vascular. Tell me one organ which is avascular in our human body. Avascular. You are close. It is cornea. Okay, cornea has got no blood supply. So that's why we are donating. Okay, so cornea. Then it is living and constantly changing. As because bone is constantly changing, there it is remodeling itself. Okay, and it is mineralized connective tissue. So it has got 2 type of minerals. I told already, calcium and phosphate. Okay. So which are the 2 hormones that will increase the calcium and decrease in the calcium? Do you know in our body? So which one will increase? Which one will decrease? Parathormone will be secreted from parathyroid gland and calcitonin is from thyroid gland. Parathormone would be always increase the calcium levels, whereas calcitonin would be decreasing the calcium levels. So these 2 you have to know about the calcium metabolism. Okay. So what are the functions? First one is it is remarkable for its hardness, okay, resilience and regenerative capacity. अगर कोई fracture हो गया bone, then it would again regenerate itself, right? It won't be the same thing, but it could regenerate by itself. Okay, so that's why we could see in many kind of fracture, if the bone is moved from that particular side, what we have to do? We have to do open surgery. Okay, and then we have to closely fix it so that it could it could be together in that particular right form. Okay, so this is known as the regenerative capacity. Okay, and any kind of fracture, this is what it is written, any kind of fracture, if it will happen, then it could regenerate by itself. Okay, so these are the structure I told. There will be intracellular matrix, there will be some cell which are embedded within them. Which are that cell? These are the osteocytes. Okay, these are the osteocytes, and this osteocyte along with the matrix is known as osteon. Okay, together this unit osteocyte plus intracellular matrix is known as osteon. So the fibrous tissue gives the bone toughness and resilience. So what do you mean by resilience? So if you try to break it in many forms, it will break or not? No. Because of this characteristic that is known as resilience. Okay? And the salt which are giving the hardness or rigidity. So whatever calcium phosphate, what is the main function? It is giving the rigidity to the bone. Okay? And it has also got the blood vessels, the nerves and the lymph nodes. Okay? So these are the main thing about the bone. So here are 3 properties. What are the physical property bone has? Okay, this is one type of tissue. So what are the property? First one is See, it is submerged in a mineral acid salt. If it is removed, what will happen? I told it is giving the rigidity. So, it will become flakes. Understood? This bone, if we remove the minerals, calcium and phosphate, what will happen? It would be flakes. It would be flexible. Okay? Then second one is decomposition. Decomposition, when it will happen? After death. Okay? So, at that time, what will happen? So, only salt remains. Intracellular matrix has gone. Only salt is there. So what will happen? It will become fragile. It will become brittle. Okay. So that is known as decomposition. Gradually it happens. Okay. Then third one is that suppose if you keep any bone in any limestone cave, right? So what will happen to that? There would be petrification. There would be one thing that is known as petrified bone. So it is due to there would be the calcium carbonate would be replacing the calcium hydroxyapatite. Okay. So it would be replacing. So these three are the properties of bone. So you have to remember decomposition, you have to remember flexicity, and then the petrification. These are the three properties of bone. Okay. Then what are the functions? This is mainly asked. Which are the function of bone? First one is protection. Protection of brain, major viscera. Then in the rib cage you could see there is heart and lungs, right? Then there is urinary bladder. Okay. So these are the structures that are preserved, and also liver. This liver, where it would be situated, do you know? Right side where? Just below the rib. So which rib are we related to rib? ये question आता है. Which ribs are related to liver? 11 and 12. 10, 11 and 12. So these ribs are protecting the liver. ये तो लिखे थे ताले?
আমাদের তো আমরা রুমে কতক্ষণ ক্লাস চলছে?
হ্যাঁ?
কতক্ষণ ক্লাস চলছে? 3টা বাজে।
3:15. Okay, so first one is about the protection. Then second one is about the framework. Suppose this framework is not there, then what will happen? Is there any shape would be formed? No. Okay, so that's why framework is very important. This framework is formed. Anybody is too much, there will be height so much better. So because of this framework. Okay. Then third one is liver. So which one would be acting as a liver? For the muscles and ligaments. Okay. So you also just remember by the keywords. Okay. What are the main functions? Then fourth one is hematopoietic. So hematopoietic means formation of blood cells. So could you tell which are the precursor cells? Physiology में थोड़ा megakaryocyte for platelets, then erythrocyte for RBC, okay, then leukocyte for the WBC. So it is also come as a curse also. Because of this bone marrow, there is a formation of, do you know any disease? Blood cancer. Yeah, same. Bone marrow cancer, and another one is multiple myeloma. Okay, so these are the disease due to bone marrow. For that, what you have to do, there would be some offspring. From that, there would be some bone marrow who could take, or you could replace the blood. This is the treatment. Okay. Then next one is for storehouse. Storehouse for this calcium and phosphate, which are required for any kind of muscle contraction. Okay. So as you know, liver also there is stored. There are so many minerals that are stored in the liver, right? Glucose and also many kind of minerals. So these are the function. If the function of bone has asked, you have to say these all. Okay. Then how many types of bone? Okay. If you see the structure of bone, there are two types of bone. One is compact bone, another one is spongy bone. So where it is situated? Suppose this is a humerus. So where you will get the compact bone? So there is two ends. These are known as epiphysis, okay, and this is known as shaft, that is known as diaphysis. Okay, understood? These ends are epiphysis, and inside that is known as diaphysis. So in the diaphysis you will get the compact bone. So what is the nature of compact bone? So it would be ivory-like. It would be ivory-like rigid. Okay, and outside covering is periosteum, and inside if you cut section, okay, of this bone, what you will see? You will see one cavity, central cavity that is going inside. So that is known as medullary cavity. Now see, this is suppose a bone, right? So outside covering is known as periosteum. So if you cut inside, you will get one medullary cavity. So inside also one covering is there. So that is known as endosteum. Understood? Inside covering is endosteum, outside covering is periosteum. In between that you will have this compact bone. Okay? So this is about the shaft. Then another type of bone is spongy bone. So where you will found? On the epiphysis, on the end. So why it is spongy? Spongy means there is so much, it is porous. Okay, so many space are in between. So why it is spongy? Because of there are numerous blood vessels. I told you all the blood vessels, the periosteal, epiphyseal, juxtaepiphyseal. Okay, only nutrient artery is going to the shaft. Remaining all are present on the to the end. Okay, so this is about the epiphysis on the both sides to the two ends. Okay, then what is the difference between two bones? How it is divided? That which one is a compact and which one is spongy. So first thing you have to say the relative amount of solid matter. So solid matter is more in compact bone. Okay. Then number and size and spaces they contain. So that is the spongy bone has more. It is porous. Okay. So by that way it would divide. And if you see this bone under microscope, then also you could see. Okay, if you cut the shaft, you will get the compact bone, and if you cut the epiphysis, there you will get the spongy bone. Okay, histology class 5. So there you could see. Okay, so see, this is I am talking about. This is the spongy bone. This is the compact bone. So inside there is medullary cavity. Okay, and what he has said, there is another structure that is known as metaphysis. Okay, so which one is the metaphysis? Between the epiphysis and diaphysis. And another thing would be here that is known as epiphyseal cartilage. Okay, I will come on the next class. Epiphyseal cartilage means it would be present over here. So what is the function of epiphyseal cartilage? It will help to lengthening the bone. Okay, this is the side where the bone used to lengthen. Okay. So again, what are the characteristics of compact bone? First one is the shaft of the body of all long bone. Whatever long bone we have got. So could you say one miniature long bone? Example? Miniature. Clavicle is a modified long bone. Modified long bone. Okay. Metacarpal and metatarsal. If you see the metacarpal and metatarsal, okay, these are the metacarpal and metatarsal, right? It is looking like a long bone only, but it is a miniature part. So it is known as miniature long bone, and modified long bone is clavicle. Okay, so these are the example. Then, if a longitudinal section is made through the long bone, it's found to contain a central cavity. I told that is known as medullary cavity. Inside that, which thing would be there? Why the medullary cavity is there? What it contains? Which type of bone marrow? Remember, this shaft contains both red and white. Whether only the epiphysis has got red bone marrow. Okay, here you will get yellow and red, but epiphysis has got red bone marrow. Okay. So externally the bone is covered by a membranous sheath, so that is known as periosteum. Okay, and internally it is covered by endosteum. and between these two layers lies a thick ivory-like substance, that is the compact bone. Okay, and it gives attachment to muscle and ligament. All the muscle and ligament would be attached over the sac on the compact bone. Okay, and remember, if muscle to act, it has to cross a joint. Okay, so that origin would be somewhere, insertion would be somewhere, and it would be crossing the particular joint. Suppose shoulder joint, so it would be taking origin from either clavicle or scapula, and it would be inserted where? On the humerus. Okay, so this is a normal rule the muscle used to follow. Okay. So see, this is a structure. So this is what I am talking about. There you will get one small minute foramina, that is known as nutrient foramina. Okay, and here you are having the epiphyseal cartilage. So many things are in the joint. Okay, there is another thing known as articular cartilage. What is that? Articular cartilage belongs to particular joint. Any joint of our body would be covered by one cartilage, that is known as articular cartilage. Okay. See, here also you are having elbow joint, so that is known as articular cartilage. Okay. So this is the nutrient artery which is going here. And you could see the spongy bone is porous. It is a typical honeycomb appearance. Okay. So spongy bone, what are the things we could see? First one is they have a thin outcell, that is known as compact bone, and network of lamellae of bone. Okay. So we will come across the detailed structure like Volkmann canal, everything on the next class. Okay. So what are the detailed structure of bone? Remember, spongy bone make up of 20% of mass of a bone. Any bone, spongy bone consists of 20% of mass of whole bone. Okay. And it contains, I told, it contains red bone marrow, which carry out the hematopoiesis. So main hematopoiesis is taking place where? On the epiphysis. Okay. And here there would be numerous trabeculae. Okay. The spongy bone is divided into numerous pores by the trabeculae or lamella. Okay, so this is a structure. See, this is a structure of spongy bone. How does it look like? So these are the trabeculae of spongy bone. So inside, what are the things you could see? There is venules, capillary, and this is the outer covering, that is the periosteum. See, everything is running over here. Okay. So this is the spongy bone, trabeculae of spongy bone. Okay. This is also, so see how many blood vessels are running. So this is our general idea that bone has got no blood supply, but no, it is highly vascular. Okay. So that's why if you see any road traffic accident, main blood is coming from the bone. There is tremendous bleeding and there is tremendous pain. Okay. Pain why it is coming due to nerve involvement and blood due to these blood vessels. Okay. So types of bone. What are the types? There are 5 types of bone. First one, long bone. Okay. So long bone. Which are the long bone? Femur, tibia, fibula, right? Then here humerus, radius, ulna. And this horizontally placed long bone is clavicle. And I told miniature type of long bone, metatarsal, metacarpal. Right? And there are, which are the other bone? Flat bone. Flat bone is scapula, then all these skull bone. Remember, all these skull bone are flat bone, right? Then next comes the irregular bone. Which one is the irregular bone? Hip bone is one type of irregular bone. Any other? Vertebra. Okay, vertebra also irregular bone. Then there is sesamoid bone. Example is patella. Any other example? Rider's bone. Then pisiform. So what is the typical feature of sesamoid bone? It develops from a tendon of muscle. Okay, patella, which muscle it will develop? That is quadriceps femoris. Okay, he told about the rider's bone. So that is from the adductor longus. Okay, Then another one is pisiform. Pisiform is flexor carpi armalis. Okay, flexor, because it is present on this side, on the little finger. So there will be which tendon? Flexor carpi armalis. ये MCQ आता है, right? So these are the sesamoid bone and it develops from this particular tendon. Then another type of bone, short bone. So which are the short bone? Remember, short bone would be having, there will be 6 surfaces. 6 surfaces means cubical in structure. So there will be anterior, posterior, medial, lateral, superior, inferior. Among them, 4 surfaces will be articular. Understood? Among them, 4 surfaces will be articular. That means articulating with another bone. And 2 surfaces would be non-articular. Okay. So these are the bones. So see, these are the example. First one is vertebra, right? It is irregular bone. This is the short bone. So which are the short bone? These are the tarsal bone. So see, this is the calcaneum. This is the talus. Then this one will be navicular, cuboid. Then medial, intermediate, lateral, cuboid. Okay. So there will be 3 rows on the feet. Okay. And which are the other bone? Like sesamoid bone is patella. Then long bone is femur. And then flat bone, another example is sternum. Sternum also flat bone. So again, this is another example. See all the flat bone present on the skull. So these are also known as sutural bone because they are making so many sutures in between. Okay. So long bone example is humerus, radius, ulna, femur, tibia, fibula and tibia. Okay. So these are the long bone. So example of long bone are these. Short bone. So I told they have, it is looking like a cube and remember it has got 6 surfaces, among them 4 are articular, 2 are non-articular. Examples are the carpal and tarsal bones. Okay, then flat bones. So what would be the typical feature of flat bone? It has got it is spongy bone or compact bone? Actually, it is spongy bone, but it is covered by a thin layer of compact bone. Okay, so which are the examples? Scapula, then sternum, then. So this is the, remember this is another question. These are the site for the bone marrow aspiration because the red marrow are abundant. So these are the site for bone marrow aspiration. So which are the site? Sternum. Another one, remember iliac crest. ये भी MCQ आता है. Okay, which are the commonest site for bone marrow aspiration? One is iliac crest, another one is sternum. Okay. So these are the example: sternum and the scapula, ribs and the skull bone. Ribs also example of flat bones. Okay. Irregular bone. So it has got irregular in shape, so that's why it is called. So which are the irregular bone? First one is the vertebra, then the sacrum. Sacrum is also irregular, right? And the hip bone. These are the example of irregular bones. Then sesamoid bone, patella, pisiform and ribos bone. These are the example of sesamoid bone. So why? What is the function of sesamoid bone? Why it is present? So it protects the tendon from any kind of wear and tear. So these are the particular space where there are so many movements. Okay, in the knee also continuous flexion and extension during walking, during sitting. Okay, every time. So there will be, it will be protecting the tendon for wear and tear movement. Okay, and this is also known as kneecap. So this is the example patella. So you could see the tendon. This is the tendon of quadriceps femoris. Why it is called quadriceps? There are 4 muscles altogether which are present on the anterior thigh. Okay, and they are inserting well to the patella. And this particular ligament is known as ligamentum patelli. Okay, there are 4 muscles. What are they? First one is rectus femoris, vastus lateralis, vastus medialis, and vastus intermedius. Okay, so this is known as quadriceps femoris. So that is all about today's class. Tomorrow we will be discussing about bone in details. Okay, thank you.
All of you must be acquainted with a term. That term is very important, and that term is a reference. Huh? This reference will help you in holding the bones in proper position, in holding the organs in proper position. So that term for reference or term of reference is anatomical position. Because this is a very important term, anatomical position. By this term, anatomical position, we mean a person standing with his legs closed, ঠিক আছে, দুটো পাকে কাছাকাছি করতে হবে, feet approximate, standing on the ground, looking forwards. A man standing upon the ground, looking forwards, with his feet approximated, and the upper limbs in a position like this. Upper limbs usually act like this, right? আমরা এরকম ভাবে upper limbটাকে রাখি। But in the anatomical position, the upper limbs should be like this. এইরকম রাখতে হবে। ঠিক আছে? তাহলে this is the anatomical position. Now with this anatomical position as a reference, we will describe the bones and all the organs. All the bones and all the organs will be discussed with this anatomical position, this reference. Meaning, whenever you are asked to hold a bone, you have to assume that the bone belongs to you. ঠিক আছে? তোমার হাতে যখন আমি কোনো একটা বোন ধরাচ্ছি, ধরো say for instance this bone of the arm, humerus, or this bone of the lower limb, the femur. Whenever I will ask you to take the femur from the table, or the humerus, or for that matter radius, ulna, any bone, তোমাকে যখন হাতে নিতে বলবো, next question will be, tell me the side to which this bone belongs. First of all, you have to identify the bone. তুমি বললে স্যার this is femur, or madam this is femur. Then you will be asked to which side the bone belongs. ঠিক আছে? Whenever I ask you the side, you have to hold the bone in proper position. Otherwise, you cannot say the side. তাই না? বোনটাকে ঠিকঠাক না ধরতে পারলে তো হবে না। তাহলে বোনটাকে কিভাবে ধরতে হবে? প্রপার পজিশন মানে বোন should be held in anatomical position. That means you have to assume that the bone belongs to you. ঠিক আছে? তাহলে to make that thing as if the bone belongs to you, what you have to know? You have to know certain pertinent features regarding the bone. ঠিক আছে? Some pertinent features about the bone you have to know. Otherwise, how can you determine the site? Assuming initially that it belongs to you. ঠিক আছে? তাহলে তোমার ধরো বোনটা তোমার, femur is yours. তাহলে it may be of right or left side. তাহলে if I ask you to determine the side, you have to know certain facts about the femur. সেই facts গুলো কি? Facts গুলো হচ্ছে upper and lower end. ঠিক আছে? Upper end will have certain features which the lower end will not present or will not show. Similarly, lower end will have certain features which the upper end will not show. That is, there are points to differentiate between the upper end and the lower end. By such points, you can readily determine which end of the bone should look upwards and which end of the bone should look down. ঠিক আছে? Similarly, there will be features which will help you in identifying the front of the bone from the back. তাহলে ব্যাকে কিছু ফিচার্স থাকবে, ফ্রন্টে কিছু ফিচার্স থাকবে। Such points will help you to hold the bone in such a manner that the features marking the anterior part will be looking forwards. The features marking the posterior part will be looking back. ঠিক আছে? Again, there will be certain features which will be more towards the midline, and there will be certain features which will be away from the midline. More towards the midline, the features, এটাকে আমরা বলি medial, ঠিক আছে? Away from the midline, we call it as lateral. তাহলে in a bone, in a long bone, এই যে বড় বড় বোন আছে অথবা in short bones also, you will find these features, upper end, lower end, assuming that the person is in anatomical position, দাঁড়িয়ে আছে। কারণ শুয়ে পড়লে দেখো আপার এন্ড লোয়ার এন্ড তো থাকছে না, তাই না? শুয়ে পড়লে কি হবে? হেড এন্ড টেল, যে একটা হেডের দিকে সেটা হেড এন্ড, সেটাকে তো আপার বলতে পারবো, তুমি তো শুয়ে আছো টেবিলের উপর, তাই না? ইউ আর অন দ্য টেবল, তাহলে ওয়ান অফ দ্য এন্ডস উইল বি হেড এন্ড, দ্য আদার ওয়ান উইল বি টেল এন্ড। তো আমরা হেড এন্ড টেল এন্ড বলি না, সিন্স উই হিউম্যানস আর বাইপেডস, আমরা যেহেতু বাইপেডস সেই জন্য আমাদের হেড এন্ড টেল এন্ড পড়ার দরকার হয়, আমরা আপার এন্ড লোয়ার এন্ড বলেই বলবো। তাহলে আপার এন্ড, লোয়ার এন্ড, anterior, posterior, features over the front, features over the back, then features over that aspect of the bone which is more towards the midline, medial features, lateral features. ঠিক আছে? আচ্ছা, তাহলে বোনস এর ক্ষেত্রে ধরো এরকম। এবার আমি একটা লং বোনের কথা বলি। বোনটা তো ধরো একটা রিব হতে পারে না, এই যে রিবস আছে thoracic skeleton-এ, এই যে রিবস আছে, আচ্ছা এই রিবস গুলো কিরকম? কার্ভড, তাই না? রিবস আর কার্ভড, ফ্ল্যাট, ঠিক আছে? ফ্ল্যাট রিবস আর কার্ভড, ফ্ল্যাট। তাহলে রিবস কে কিভাবে আমরা ডেসক্রাইব করি? পিছনে কি আছে? thoracic part of the vertebral column, thoracic column. The ribs make joints with the thoracic part of the vertebral column. তাহলে that end of the rib which meets the thoracic part of the vertebral column is lying behind, তাহলে that must be the posterior end, ঠিক আছে? The end that looks in front, that is in front, এগুলো হচ্ছে costal cartilage, ঠিক আছে? এগুলো costal cartilage, these are the ribs. তাহলে the end which is in front making joints with the costal cartilages, that end is the anterior end. Now, the anterior end will have certain features which will be missing at the posterior end. Similarly, posterior end will have certain features which will be missing in the anterior end. Now, by such features, one can easily differentiate between the posterior and anterior end. তাহলে একটা রিব তোমার হাতে যখনই আমি দেব, তোমাকে ফার্স্ট অফ অল কি করতে হবে? দুটো এন্ড ডিফারেন্সিয়েট, which one is anterior and which one is posterior. Otherwise, তুমি উল্টো করে ধরবে। রিবটাকে শুধু এরকম ধরলে তো হবে না, তুমি পেছনটাকে সামনে দিয়ে দিলে সব গন্ডগোল হয়ে যাবে। নেক্সট কি? এই যে রিবটা দেখো, এর একটা উপরে একটা বর্ডার আছে, আবার নিচে একটা বর্ডার আছে, ঠিক আছে? তাহলে একটা আপার বর্ডার আছে, একটা লোয়ার বর্ডার আছে। তাহলে মার্কিং দ্য আপার বর্ডার, there will be certain features, or there will be no features at the, there will be no salient features at the upper border, but there will be features along the lower border. তাহলে such features along the lower border will help you to differentiate between the upper border and the lower border. একটা বর্ডারে কোনো ফিচার্স নেই, ঠিক আছে? আর একটা বর্ডারে ফিচার্স আছে। তাহলে the border with features will be the lower end, or sorry, will be the lower border. The border without features is the upper border. ঠিক আছে? আচ্ছা, এইবার রিবসটা অত সহজ নয়, পরে যখন আসবে তখন আমরা দেখো বলবো রিবের পোস্টেরিয়র এন্ড, সেখানে অনেক কিছু আছে, কি কি পার্টস আছে সেটা আমরা, কিন্তু এটা বেসিক জিনিস। এবার নেক্সট দেখো, এই যে রিবটা, এর একটা দিক, মানে ব্যাপারটা কি, এইটাই হচ্ছে অ্যানাটমি, বুঝলে? মানে তুমি যতদিন পর্যন্ত অ্যানাটমি করোনি তখন তোমার কাছে রিব মানে রিব। বায়োলজিতে কি পড়েছো? রিবস, there are 12 pairs of ribs, ঠিক আছে, in the human skeleton, that's all. কিন্তু তুমি যখন ডাক্তারি করবে তখন তোমাকে বাইরে ভেতরে সব বুঝতে হবে, কারণ রিবসের ভেতরে কি আছে? রিবস along with thoracic part of the column and the sternum make up the thoracic cage. এবার এই যে thoracic cage ইনকমপ্লিট, cage তো খাঁচা, ঠিক আছে? খাঁচার স্কেলেটাল ফ্রেম, ঠিক আছে? Osseo cartilaginous frame হচ্ছে ribs, costal cartilages, sternum, and behind the thoracic part of the column. 12 thoracic vertebrae are there. Pairs not. 12 thoracic vertebrae are there across the dorsal midline. Direct thoracic cage. The incompleteness in the cage will be reached over by the muscles. মাপ রাখলে তো হবে না। তাহলে between the ribs muscles আছে, intercostal muscles গুলি। তবে thoracic cage-টা কমপ্লিট হবে। ঢাকা পড়বে। এবার ভেতরে লাংস আছে, with the pleura, হার্ট আছে। তাহলে ভেতর আর বাইরে তো কোনোদিন এক হতে পারে না। বুঝেছো? বাইরে তো আমরা লাংস হার্ট পাচ্ছি, thoracic cage-এর ভেতরেই আমরা লাংস হার্টকে পাচ্ছি। তাহলে ribs এর একটা outer surface আছে, meant ribs, all the ribs have got an outer surface meant for some function, and it has got an inner surface with which the lungs, mainly the lungs, will be related. তাহলে inner features এ আমরা lungs কে দেখতে পাবো, covered by pleura. বাইরে আমরা lungs কে তো দেখতে পাবো না, heart কেও দেখতে পাবো না। তাহলে বাইরে কাকে দেখতে পাবো? Muscles. There will be muscles attached to the ribs. Truncal muscles আছে, এছাড়া truncal muscles ছাড়া upper limb এর muscles আছে ribs এ। সে যখন আমরা ribs পড়াবো তখন বলবো। তাহলে in that way every bone has to be dead. Bone মানে bone তা কিন্তু নয়, bone বলতে গেলে অনেক পড়তে হবে কিন্তু anatomy। একটা rib যখন আমি বলবো দেখবে এই যে 12 layers of ribs আছে, মানে ব্যাপারটা এখন থেকেই জেনে রাখো। ব্যাপারটা কিন্তু খুব সহজ নয়, বেশ কঠিন জিনিস। বুঝেছো? 12 pairs of ribs আছে in the human skeleton. 12 pairs of ribs. Now these 12 pairs of ribs we can classify in various manners. অনেক রকম ভাবে আমরা ক্লাসিফাই করতে পারি। Some of the ribs are being called as true ribs. Others are being called as false ribs. তাহলে which ribs are being called as true and which ones will be called as false? The true ribs are called so because false ribs are called so because reasons are there. Why we call some of the ribs as true and some of the ribs as false? Follow. Next classification is there. According to the articulation that is being made by the ribs, আমরা পেছনে কি বললাম? ভার্টিব্রাল কলাম আছে, ঠিক আছে? সামনে কাকে বলছি? স্টার্নাম আছে। Not all the ribs join with the sternum. Some of the ribs, the upper ribs, they join with the sternum, upper 7 pairs. That they have got a name of their own. Since they join with the sternum in front, the vertebral column behind, they are called as the vertebrosternal ribs. ঠিক আছে? Below the 7th pair, a few pairs below the 7th pair will be joining with each other, not with the sternum. ঠিক আছে? তাহলে সামনে কে আছে? costal cartilage আছে। তাহলে between the costal cartilages we will find joints. Such ribs behind they will articulate with the thoracic part of the column. তাহলে they will be called as vertebrochondral ribs. 11th and 12th pairs, they do not join with anything. They remain free. Therefore, they are being called as the floating ribs. That in that way, we can classify the ribs. ঠিক আছে? তাহলে এই কথাগুলো আমরা যখন ribs পড়াবো তখন আমরা বলবো, আবার বলা হবে। তার পাশাপাশি প্রত্যেকটা ribs নিয়ে আবার তোমাকে আলাদা করে জানতে হবে। There will be some pairs which will have common features. Such ribs will be called as typical ribs. ঠিক আছে? তার মানে কি? They will have a common type, যার জন্য আমরা তাকে বলছি typical. There will be other ribs with special features. তাদের আমরা বলবো atypical ribs. তাহলে which ribs are atypical and what are those special features? You have to know. তাহলে তুমি ভাবো এই ribs নিয়ে তোমাকে এত কিছু জানতে হবে। এবার ribs নিয়ে তুমি দেখবে আমরা কটা ক্লাস নিই পরে, দেখবে অলটুগেদার 3টে ক্লাস আমাদের হয়তো ribs নিয়েই নিতে হবে, অস্টিওলজি। মিনিমাম 2টো, ম্যাক্সিমাম 3টে। তাহলে in that way we will deal with the bones. প্রত্যেকটা bones নিয়ে দেখবে কত কথা তোমাকে জানতে হবে to be an MBBS doctor. বুঝছো? এমনি এমনি তো হবে না, হাওয়ায় হাওয়ায় এমবিবিএস হয়ে যাবে না। তাহলে এখান থেকে শুরু হচ্ছে, অ্যানাটমিতে প্রচুর পড়তে হবে, প্রচুর। আচ্ছা যাই হোক, ম্যাডাম তো তোমাকে অ্যাপেন্ডিকুলার স্কেলেটন, অ্যাক্সিয়াল স্কেলেটন বলেইছে। আচ্ছা, অ্যানাটমিক্যাল পজিশন টার্মটা তুমি শুনলে ইন রিলেশন টু বোনস। এবার ধরো আমার কাছে এখানে বোনস নেই, আমি চলে আসি, আমি নিই বোনস গুলো নিয়ে কালকে যারা করবে ক্লাস নেবে আমি বলবো সবাইকে বোনস নিয়ে আসতে, ছোট ছোট বোনস আলাদা, ডিসআর্টিকুলেটেড বোনস নিয়ে আসতে। তুমি দেখো এই যে হাতে যে বোনস গুলো আছে, এই যে এখানে, ঠিক আছে, এটা হচ্ছে হ্যান্ড প্লেট, তাই না? সেরকম ভাবে ফুট প্লেট আছে। তাহলে ফুট প্লেটে যে স্কেলেটন আছে ওটাকে আমরা বলবো স্কেলেটন অফ দ্য ফুট অর পেডাল স্কেলেটন, ঠিক আছে? এখানে স্কেলেটন অফ দ্য হ্যান্ড আছে। এবার স্কেলেটন অফ দ্য হ্যান্ডটা তুমি দেখো, যদি দেখো এখানে লম্বা লম্বা এরকম বোন কিন্তু নেই, ঠিক আছে? প্রথমে কতগুলো ছোট ছোট বোন আছে, স্মল বোনস আছে। তারপরে এরই মিনিয়েচার সংস্করণ আছে, ছোট সংস্করণ। দেখো এগুলো ছোট সংস্করণ, এগুলো আরো ছোট সংস্করণ, লম্বা কিন্তু এর মতোই দেখতে কিন্তু অনেক ছোট। সেই জন্য এদেরকে আমরা কি বলবো? এদেরকে আমরা বলবো মিনিয়েচার লং বোনস অর শর্ট লং বোনস, ঠিক আছে? তাহলে শর্ট লং বোনস এর এক্সাম্পলস কি? মেটাকার্পাস, ফ্যালাঞ্জিস। পায়ে যখন যাব মেটাকার্পাস ওখানে বলবো না, ওখানে বলবো মেটাকার্পাস। কিন্তু টোয়ের যে বোনগুলো আছে একই নাম, ফ্যালাঞ্জিস, ঠিক আছে? ফ্যালাঞ্জিসের নাম কি করে বলতে হয় ম্যাডাম হয়তো পড়িয়েছে, হ্যাঁ? প্রক্সিমাল, মিডল, ডিস্টাল, ঠিক আছে? প্রক্সিমাল, মিডল, ডিস্টাল। এবার এই যে ছোট ছোট বোনগুলো ম্যাডাম নাম বলেছে নিশ্চয়ই, কি নাম বলেছে বলো? আর বলেন না না না, ওগুলো নাম as a whole এই যে স্মল বোনস they are being called। তোমাকে যখনই ধরো পেডাল স্কেলেটন বা স্কেলেটন অফ দ্য হ্যান্ড বলতে বলা হবে, তোমাকে প্রথমে বলতে হবে হ্যান্ড প্লেট। হ্যান্ড প্লেট, the bones that we find are classified into three groups. The most proximal ones are called as carpals, ঠিক আছে? The next series in line will be called as metacarpals, and finally there will be the phalanges making up the digits of the fingers. তাহলে এখানে কারা আছে? এখানে আছে phalanges, এখানে কি আছে? এখানে আছে metacarpals, আর একেবারে most proximal goes to the list carpals, ঠিক আছে? carpals or carpus আমরা বলি। তাহলে carpal bones ও এই মাত্র নামগুলো বলছিল, carpal bones আর 18 নাম, ঠিক আছে? 8টা আছে। এবারে এই যে 8টা তোমাকে যখন বলতে হবে তখন তোমাকে in sequence বলতে হবে। প্রথমে কি? carpals are organized in two rows, a proximal row and a distal row. তুমি যদি close inspection of the carpals তুমি যদি করো তাহলে দেখবে একটা কার্ভড প্রক্সিমাল রো আছে প্রথমে, তারপরে আরেকটা কার্ভড ডিস্টাল রো আছে, বুঝেছো? এরকম আর্কের মতো আছে, একটা প্রক্সিমাল রো অফ কার্পাস এন্ড এ ডিস্টাল রো অফ কার্পাস। এবার এই যে দুটো আর্ক আছে, প্রক্সিমাল রো, ডিস্টাল রো, প্রত্যেকটা 4টে করে বোন আছে। সেই 4টে বোন ইন সিকোয়েন্স বলতে হয়। সেটা ও বলছিল, ওরকম আমি বলে দিতে পারি, কিন্তু দরকার নেই এক্ষুনি বলা, ঠিক আছে? তাহলে কার্পাস 8টা আছে। টার্সাল তুমি পায়ে দেখো, পেডাল স্কেলেটনটা দেখো, এই মানুষটার পেডাল স্কেলেটন দেখো, কিন্তু এক্সটেন্ডার করতে পারবো না, ফ্লেক্স করতে হবে। ফ্লেক্স করে এরকম দেখা, এই পেডাল স্কেলেটন পায়ে গ্রাউন্ডের উপর থাকবে। এবার এই পেডাল স্কেলেটনে আমরা যখন দেখব তখন দেখব অ্যাঙ্কেল জয়েন্টের কাছে কতগুলো বড় বড় রয়েছে, কার্পালসের মতো ছোট ছোট নয় কি? মানে কার্পালস কেন বড়, ছোট ছোট, টার্সালস কেন বড়? সহজ, ঠিক আছে? আমাদের ইন হিউম্যান, সিন্স উই স্ট্যান্ড অন আওয়ার লেগস, ঠিক আছে, ফিট আর ওয়েট বিয়ারিং, ফিট আর ওয়েট বিয়ারিং। এবার আমার ওজন ধরো কম, 52-53, আমি জানি না ওজন কি অনেক, 100 কেজি 110 কেজি হতে পারে মানুষ, তারও বেশি হতে পারে, এক্সেপশন আছে। তাহলে 110 কেজির একটা মানুষ, ঠিক আছে, 110 কেজি উইল বি বোর্ড বাই হিজ ফিট অর হার ফিট। মেয়েরাও মোটা হয়, হ্যাঁ, তাই শুধু হিজ বলবো না, হারও বলতে পারি, 120 কেজি হতে পারে, হ্যাঁ, 1 কুইন্টালের বেশি। তাহলে the entire weight will be borne by the feet. তাহলে পেডাল স্কেলেটনের বোনস গুলো obviously they will be heavier then the bones are hanging. এটাই হচ্ছে সিম্পল রিজন, বুঝেছো? ওয়েট বিয়ারিং যেহেতু pedal skeleton, since it is weight bearing, therefore tarsal bones will be much larger and heavier, ঠিক আছে, or heavily built than the carpus. homologous কিন্তু একই রকম, কিন্তু ওগুলো অনেক বড়। Moreover, since we stand on our legs, the tarsals are organized in a different manner than the carpus. হাতে যেরকম আছে একটা proximal row in an arch form and a distal row in an arch form, আমরা কিন্তু পায়ে তা দেখতে পাই। পায়ে কি দেখবো? টার্সাল বোনস 7টা আছে, এখানে 8টা আছে, 8টার ভিতরে একটা অবশ্য ছোট একটা বনি নডিউল পিসি ফর্ম, তুমি বাকিগুলো 7টাই ধরতে পারো। তো পায়ে আমাদের 7টা আছে, 7টার ভিতরে the bone which forms the ankle is the talus, ওই বোনটার নাম হচ্ছে talus। তাহলে অ্যাঙ্কেল জয়েন্ট যে বোনটা পড়ছে তার নাম talus। অ্যাঙ্কেল জয়েন্ট মানে কি? উপরে কি আছে? টিবিয়া এন্ড ফিবুলা, বোনস অফ দ্য লেগ। নিচে কি আছে? অনলি talus, আর কিন্তু কেউ নেই। কিন্তু তুমি যখন হাত পড়বে, হাতের যখন বোনস পড়বে, তখন তুমি দেখবে যে প্রক্সিমাল রুটে যে 3টে বোন আছে, 3টে বোনই কন্ট্রিবিউট করে কিন্তু ইন দ্য রিস্ট জয়েন্ট, ঠিক আছে? 3টে বোনই কন্ট্রিবিউট করে, ঠিক আছে? নট ইন অল পজিশনস অফ দ্য রিস্ট, কিন্তু 3টে বোনের কন্ট্রিবিউশন আছে। নট দ্য ফোর্থ ওয়ানটা একটা ছোট পিসি ফর্ম, ওটার ফাংশনও অন্যরকম, সেই ওটা কন্ট্রিবিউট করে, কিন্তু রেস্ট উইল বি কন্ট্রিবিউটিং in the formation of wrist. But in case of leg or lower leg, the only bone that will contribute to the ankle homologous with the wrist. Wristed সঙ্গে homologous, কারণ wristed radius আলনা আছে, bones of the forearm, তার সঙ্গে তাহলে 3টে বোন, scaphoid, lunate, triquetral, এই 3টে বোনই articulate। সেই জন্য wrist জয়েন্টের অ্যানাটমিক্যাল নাম কি? রেডিওকার্পাল জয়েন্ট, বুঝেছো? রেডিওকার্পাল জয়েন্ট। আলনা কিন্তু নেই আমার। দেখো এই যে রিস্ট, এই যে রিস্টটা দেখছো, এই রিস্ট হচ্ছে অ্যাঙ্কেলের হোমোলোগাস। পায়ে যে অ্যাঙ্কেল আছে তার হোমোলোগাস। পায়ে কি? বোথ বোনস অফ দ্য লেগ কন্ট্রিবিউটিং, টিবিয়া কন্ট্রিবিউট করছে, ফিবুলা কন্ট্রিবিউট করছে। কিন্তু যখন আমি পেডাল স্কেলেটনে আসছি, তখন একটাই বোন কন্ট্রিবিউট করছে, সে কি? টেলাস। সেই জন্য অ্যাঙ্কেল জয়েন্টের নাম হচ্ছে টেলোডুরাল জয়েন্ট, বুঝেছো? গ্রাস মানে লেগ, বুঝেছো? গ্রাস মানে লেগ, তাহলে লেগের দুটো বোন টিবিয়া ফিবুলা বোথ কন্ট্রিবিউট, সেই জন্য এর নাম হচ্ছে টেলোডুরাল জয়েন্ট। সেরকম ভাবে রিস্ট জয়েন্টের নাম কি? রিস্ট জয়েন্ট যে কেউ বলবে এটাকে কবজি বা রিস্ট, ঠিক আছে? ইংরেজরা রিস্ট বলবে, আমাদের বাঙালিরা কবজি বলবে, একই জিনিস, কিন্তু এটা হচ্ছে লেম্যানস টার্ম। তুমি যে মুহূর্তে ডাক্তার হচ্ছ, তোমার কিন্তু নামটা মনে রাখতে হবে। নামটা কি? রেডিওকার্পাল জয়েন্ট। তার কারণ উপরে কেবলমাত্র রেডিয়াস আছে, তুমি নাম করতে পারছো, নিচে কিন্তু কোনো একটা বোন নেই, সেই জন্য তোমাকে রেডিওকার্পাল বলতে হচ্ছে। পায়ের ক্ষেত্রে কি ছিল? নিচে একটা বোন ছিল, সেই জন্য তুমি টেলাসের টেলো বলবে, কিন্তু উপরে দুটো বোন আছে, বড় নাম হয়ে যাবে টেলো টিবিউলার, বাজে শোনায়, সেই জন্য আমরা কি বলবো? টেলোডুরাল জয়েন্ট, অ্যানাটমিক্যাল নাম, বুঝেছো? তাহলে এইগুলো হচ্ছে অ্যানাটমি জানবে, শুধু নামের জানবে। আমি শুধুই তোমাকে পরীক্ষায় এই নাম দিয়ে জিজ্ঞেস করি, খাওয়া করে, বুঝেছো? মানে নামের জানবে সব তুমি স্ট্রাকচার মানেই তার নাম থাকবে, এটা কি ওটা কি, এই নাম, বুঝেছো? এবার নামেই তুমি আউট হয়ে যাবে। আর কত নাম মনে রাখবে বলো না? এই তো তোমাকে আমি একটা দুটো নাম বলছি। যাই হোক, তাহলে পায়ের ক্ষেত্রে আমরা কি বলছি? যে টার্সালস আর নট অর্গানাইজড অ্যাজ দ্য কার্পাস, তো বোথ দ্য গ্রুপস আর হোমোলোগাস। ওটা পায়ে আছে, এটা হাতে আছে, একই রকম ফাংশন তো একসময় সার্ভ করতো। আমরা তো 4টে লিম্ব দিয়ে কোয়াড্রিপেডরা হাঁটে, তাই একই রকম ফাংশন সার্ভ করে, কিন্তু মানুষের ক্ষেত্রে অন্যরকম হয়ে গেছে, যেহেতু আমরা দু পায়ে দাঁড়িয়েছি, সমস্যা বেড়েছে। কমে তো নেই, উল্টে সমস্যা বেড়েছে দু পায়ে দাঁড়ানো, বুঝেছো? আবার তারপরেই মানুষ, ঠিক আছে? মেসি আর এমবাপের মতো ফুটবল খেলতে পারবে না, বুঝেছো? মানুষ ফুটবল খেলছে, তোমাকে আনন্দ দিচ্ছে, বুঝেছো? এত বড় এন্টারটেইনমেন্ট ইন্ডাস্ট্রি, জিও সিনেমা সারা পৃথিবীতে এগিয়ে নিল, হ্যাঁ? না সবই ইন্টাররিলেটেড, বুঝেছো? মানুষ তার বুদ্ধি লাগিয়ে সবকিছু হয়ে আছে, তারপরে রোজগার সবকিছু। যাই হোক, তাহলে টার্সালস আর অর্গানাইজড ইন এ ডিফারেন্ট ম্যানার দ্যান দ্য কার্পস। তাহলে টার্সালস এর টেলাসটা এগুলো আর্টিকুলেট। এইবার দেখো টেলাস ফেলস টু রিচ দ্য গ্রাউন্ড। মজার ব্যাপার হচ্ছে টেলাস আমি রেখেছি একটা জয়েন্ট করেছে, কিন্তু ও কিন্তু গ্রাউন্ডকে টাচ করতে পারছে। নেচার কি জুডিশিয়াস দেখো ওর অ্যাপ্রোচে, যখন তোমার শরীরটা বানাচ্ছে টেলাস ইজ নট রিচিং। টেলাসে বডি ওয়েটটা এলো, কিন্তু টেলাস কিন্তু নিচে গ্রাউন্ডকে কানেক্ট করবে। কানেক্ট করছে কি টেলাসের নিচে যে বোনটা বসে আছে ক্যালকেট। কাজে উপর থেকে ঝাঁপ দিতে গিয়ে তুমি যদি পড়ো, ধরো যে কোনো একটা অ্যাটেক তোমাকে ও ফেলে দিচ্ছে, হ্যাঁ, বাড়ির উপর থেকে, অথবা তুমি নিজেই ঝাঁপ মারছো, ঝাঁপ মারো পায়ের উপর যদি পড়ো, ক্যালকেনিয়ামটা ফ্র্যাকচার হয়ে যাবে, বুঝেছো? তার কারণ ক্যালকেনিয়াম ইজ টাচিং দ্য গ্রাউন্ড। হিল মানে ক্যালকেনিয়াম ইজ দ্য হিল। তাহলে ক্যালকেনিয়াম ইজ টাচিং দ্য গ্রাউন্ড, নট দ্য টেলাস। তাহলে টেলাস ওয়েটটা পাচ্ছে, পেয়ে এই ওয়েটটা ডিস্ট্রিবিউট করবে। 50% অফ দ্য ওয়েট ক্যালকেনিয়াম বিয়ার করবে, ঠিক আছে? 50% will move forwards, ঠিক আছে? Walls of the toes will bear 50% of the body. যাই হোক, সেটা যখন আমরা ওয়েট ট্রান্সমিশন পড়াবো পায়ের রিলেশনে, তখন বলবো, তুমি পড়ান না কেন সব কথা বলবো। তাহলে টার্সালস গুলো কিন্তু অন্যরকম। তার মধ্যে টেলাস ক্যালকেনিয়াস হেভি। টেলাস ক্যালকেনিয়ামের সামনে যে বোনগুলো আছে তারা আবার অর্গানাইজড আছে দুটো রোতে, একটা ইন্টারমিডিয়েট রো আছে, একটা সিঙ্গেল বোন আছে। ইন্টারমিডিয়েট রোতে ন্যাভিকুলার বোর্ডছে, তার সামনে পরপর সাজানো আছে চারটে বোন পায়ের, তাহলে এই হাতের। তাহলে ডিস্টাল রো অফ টার্সাসে কিন্তু আমরা কার্পাসে যেরকম দেখেছি ডিস্টাল রোতে চারটে বোন, এখানেও চারটে বোন দেখতে পাবো, কিন্তু প্রক্সিমালি কিন্তু ওই রকম অর্গানাইজেশন হয়, কমপ্লিটলি সেপারেট অর্গানাইজেশন ইন দ্য পেডাল স্কেল, ঠিক আছে? আচ্ছা, এইবার কথা হচ্ছে ধরো যে কোনো একটা বোন, বড় বোন বা ছোট বোন, এই যে স্কেফয়েড, ট্রাইকোয়েট্রাম, ছোট ছোট বোন, এই বোনগুলোর ডিটেইলস কি করতে হবে? এখনকার এই 13 মাসের সিলেবাসে তোমাকে পড়তে হবে, কিন্তু কোনো এক সময় পড়তে কিন্তু সবগুলোই, বুঝেছো? ওই ছোট্ট বোনটা নিয়ে ওর 6টা সারফেস আছে, বুঝেছো? এই দেখো অ্যানাটমিক্যাল পজিশন। তাহলে বোনগুলো তো এখানে আছে, কার্পাস এখানে আছে, ঠিক আছে? তাহলে এই বোনের একটা সারফেস আছে লুকিং ফরওয়ার্ডস, ঠিক আছে? এই যে সারফেসটা যেটা লুকিং ফরওয়ার্ড, সেটা কোথায় আছে? পামে আছে, পাম। হ্যান্ড প্লেটের এই জায়গাটাকে তো পাম বলি। তাহলে যে সারফেসটা লুকিং ফরওয়ার্ডস হবে সেটাকে আমরা বলবো পামার সারফেস। অপোজিট সারফেসটাকে বলবো ডরসাল সারফেস, ঠিক আছে? অপোজিট সারফেসটাকে ডরসাল সারফেস হলো। তাহলে একটা পামার সারফেস হলো, একটা ডরসাল সারফেস। 6টা সারফেস থাকবে। এই ছোট্ট বোন 6টা সারফেস। যখন আমরা পড়াতাম 1.5 বছরের কোর্সে, প্রত্যেকটা বোন আলাদা করে আমাদের পড়াতে হতো, 6টা সারফেস। তারপরে দেখো এখানে যে বোনগুলো সাজানো আছে 3টে প্রক্সিমাল রোতে, ভাবো স্কেফয়েড, রুলেট, ট্রাইকোয়েট্রাম। এদের একটা সারফেস আছে টুয়ার্ডস দ্য রিস্ট, আর একটা আছে অ্যাওয়ে ফ্রম দ্য রিস্ট। তাহলে টুয়ার্ডস দ্য রিস্ট যেটা আছে সেটা প্রক্সিমাল সারফেস, যেটা অ্যাওয়ে ফ্রম দ্য রিস্ট আছে ডিস্টাল সারফেস। ইন দিস ওয়ে উই গিভ নেমস। একটা অ্যান্টোনিম হবে আর, বুঝেছো? একটা যদি পামার হয় তাহলে আরেকটা ডরসাল। এবার পামারটা রিয়েলি ভেন্ট্রাল সারফেস, ঠিক আছে? ভেন্ট্রাল সারফেস পামে আছে বলে আমরা পামার সারফেস বলছি, ঠিক আছে? তাহলে ভেন্ট্রাল ডরসাল আর পামার ডরসাল, প্রক্সিমাল, ডিস্টাল, চারটে হয়ে গেল। নেক্সট হচ্ছে এই যে এখানে বোনসগুলো আছে, একটা সারফেস থাকবে ক্লোজার টু দ্য মিড লাইন এদিকে, আর একটা অ্যাওয়ে ফ্রম দ্য মিড লাইন। তাহলে যেটা ক্লোজার টু দ্য মিড লাইন থাকবে সেটা মিডিয়াল সারফেস, যেটা অ্যাওয়ে ফ্রম দ্য মিড লাইন থাকবে সেটা ল্যাটারাল সারফেস। তাহলে ল্যাটারাল সারফেস, এই ছোট্ট বোন, lateral surface will have certain features which will be missing in the medial surface. Medial surface will be certain features which will be missed by the lateral surface. বুঝেছো? Similarly, palmar surface will have some features which will not be there over the dorsal surface. তাহলে in this way we differentiate between the surfaces. After differentiating between the surfaces, we can readily ascertain which surface is which one. এইটা করার পর আমি হাতের উপরে বসাবো, ডান হাত না পা। তাহলে প্রবলেমটা কত বিরাট প্রবলেম চিন্তা করতে পারছো? ওইটুকু ছোট্ট বোন, তাতে তোমাকে ফিচার্স খুঁজে খুঁজে বের করতে হবে। কিন্তু পাওয়া যায়, এমন কিছু ডিফিকাল্ট পাওয়া যায়, ঠিক আছে? সবই পাওয়া যায়, পাওয়া যাবে না কেন? সায়েন্স এতদিন ধরে চলছে, অ্যানাটমি সব বলা আছে বইয়ে। এবার পায়ের ক্ষেত্রে টেরাসটা বেশ বড় বোন, এই যে টিবিয়া ফিবিউলার নিচে যেটা বসে আছে, ক্যালকেনিয়ামটা বেশ বড় বোন। তাহলে এই টেরাস ক্যালকেনিয়ামে কিরকম সারফেস আছে, এই বোনগুলোকে কিরকম বোন বলে ম্যাডাম তো বলেছে, বলো। এগুলো একটা বিদ্যুতের মতো দেখতে, হ্যাঁ, কোনোটা একটু খুব ছোট, কোনোটা একটু বড়। এগুলোকে এক কথায় আমরা বলবো শর্ট বোনস। শর্ট বোনস, ঠিক আছে? শর্ট বোনস উইল বি ল্যাকিং অল দ্য ফিচার্স অফ এ লং। এইভাবে মনে রাখো, short bones, they are smaller in size and they will not have all those features that characterize a long. ঠিক আছে? এই যে বন্ধু পেছনে ওর ঘাড়ের ব্যায়াম করছে, হ্যাঁ বুঝতে পেরেছো? তোমাকে বলছি চশমা, হ্যাঁ না ঠিক আছে, ঘাড়ের ব্যায়াম মাঝে মাঝে করা ভালো, হ্যাঁ ব্রেনের সার্কুলেশনটা ভালো হয়, করা ভালো। তবে বেশি করতে যেও না, তাহলে কিন্তু আবার অনেক সময় চক্কর লেগে যায়, ডিজিনেস হবে, বুঝেছো? মানে আনইউজুয়াল পজিশন নেকের উপরে হেডের হলে কিন্তু ডিজিনেস হলে তুমি অজ্ঞান হয়ে যেতে পারো টেম্পোরারি। মানে ঘাড়ের ব্যায়াম বুঝে করতে হবে, হ্যাঁ সেই জন্য সেলুনে গিয়ে ম্যাসাজ নেয়, বুঝেছো? ওই ম্যাসাজ দিতে গিয়ে অনেক রকম বিপত্তি কিন্তু হতে পারে, হ্যাঁ মটমট করে আওয়াজ করে, হ্যাঁ খুব আরাম লাগে, কিন্তু ওই আরাম দিতে গিয়ে কিন্তু যদি সার্ভাইকাল স্পন্ডিলাইটিস থাকে তাহলে ভাবটা ট্যাবটা বাজবে, বুঝেছো? তুমি হাবিবসে গেলে, হাবিবসে গেলে মেসেজ খাওয়ার জন্য, এদিকে আমি যদি যাই আমার তো সার্ভাইকাল স্পন্ডিলাইটিস হয়ে বসে আছে, তবে হাবিবরা এক্সপার্ট, প্রফেশনাল, ওরা আমাকে মেসেজ দেবে না, যেটা তোমাকে, বুঝেছো? আমাকে আগে থেকে জিজ্ঞেস করবে, প্রফেশনাল পিপল টু দে নো দ্য টিডবিটস অফ देयर প্রফেশন। তো যাই হোক, এবার নেক্সট হচ্ছে পায়ের ধরন। টেরাস বা ক্যালকেনিয়াম আমি যখন বলবো তার সারফেস গুলো কিরকম? টেরাসটা এরকম রয়েছে, ধরো টিবিয়া ফিবুলার, এতে আমার কাছে এখন হাতে নেই বলে একটা সারফেস আছে which will be towards the ankle, ঠিক আছে, which will be towards the ankle and another will be away from the ankle. The surface that is away from the ankle, ঠিক আছে, is looking towards the ground, ঠিক আছে, or is looking towards the sole of the foot. পায়ের পাতাটাকে আমরা সোল বলি, হ্যাঁ এসওএল সোল, ঠিক আছে, এসওইউএল সোল নয়, আপনার সঙ্গে ঘুরিয়ে ফেললে মুশকিল হয়ে যাবে, হ্যাঁ তাহলে এসওএল সোল। এবার দুটো নিয়ে আছে, সোল আছে তুমি জানো, হ্যাঁ হাফ সোল, জানো তো এসব টার্ম আছে কিন্তু আমাদের সময় সব চায়ের দোকানে আড্ডাবাজিতে সব খুব চলতো। কি রে হাফ সোল খেয়েছিস? হাফ সোল খাওয়া মানে কি জানো? গার্লফ্রেন্ড ডিচ করেছে, হ্যাঁ এসব ছিল। চ্যাংড়াদের মধ্যে চায়ের দোকানে এরকম খাওয়া হতো, বুঝেছো? না এটা আজকাল চলে না, এগুলো আমাদের সময় চায়ের দোকানে বসি। যাই হোক, তোমাকে আমি বলি সোল মানে হাফসোল মানে বুঝতেই পারছো, সোল চেপেটা, এটা কথা, সেখান থেকে এই কথাটা উঠছে, হাফসোল না ফুল সোল। তো এইবার তোমার কথা হচ্ছে সোল এসওএলি সোল। আচ্ছা তাহলে সোলের দিকে যে সারফেসটা, তার মানে নিচে যে সারফেসটা আছে ওটাকে আমরা বলি প্লান্টার সারফেস, ঠিক আছে, আর তার উপরে যে সারফেসটা ওটাকে ডরসাল সারফেস, প্লান্টার ডরসাল। এবার পায়ের ক্ষেত্রে পা তো এইভাবে নেই, তাই না? হাতটা আমরা এইভাবে আছে, কিন্তু পাটা তো অ্যাট রাইট অ্যাঙ্গেলস এইভাবে আছে, তাহলে পায়ের ওরিয়েন্টেশনটা পুরো অন্যরকম। তুমি যখন অ্যানাটমিক্যাল পজিশনে দাঁড়াচ্ছ দুটো পাকে এরকম ভাবে দাঁড়াচ্ছ, তাই না? দুটো পা এইভাবে দাঁড়ায়, তাহলে এই সারফেসটা হচ্ছে ডরসাল সারফেস, এই সারফেসটা প্লান্টার সারফেস। এবার যে সারফেসটা অ্যাঙ্কেলের কাছে সেটা প্রক্সিমাল সারফেস, যেটা অ্যাঙ্কেল থেকে দূরে সেটা ডিস্টাল সারফেস। আর একটা মিডিয়াল সারফেস, ঠিক আছে, যেটা টুয়ার্ডস দ্য মিড লাইন, পুরোটা লেফট হ্যান্ড সারফেস। তাহলে কোনো অসুবিধা নেই, কিন্তু এমন কিছু কঠিন না, 6টা সারফেস মনে রাখা। এবং মোস্ট অফ দ্য বোনস টার্সাসি হোক বা কার্পালি হোক, তাদের আমরা 6টা সারফেস দেখতে পাই। ঠিক আছে, এক্সেপশনস আছে, এক্সেপশনস বেশি নেই, মোস্ট মেজরিটি উই হ্যাভ 6 সারফেসেস। এবার তারপরে ধরো সারফেসের ফিচারস আছে, সেটা যখন বোনস হবে তুমি জানতে পারবে আস্তে আস্তে। এগুলো প্রথম দিনের ক্লাসে ছিল, মানে ক্লাস তাও আবার অ্যাডেন্ডাম, ম্যাডাম পড়িয়ে গেছে, আমি তারপর আবার তোমাদের বোর্ড করছি। হ্যাঁ, আমি বেশিক্ষণ বোর্ড করবো না, অল্প সময় বলবো। যাই হোক, এবার আমার যেটা মনে হচ্ছে যেগুলো বলা দরকার, তাহলে অ্যানাটমিক্যাল পজিশন বোর্ডে পড়েছি, বোন সেপারেশন খানিকটা আইডিয়া হলো। এইবার ধরো অর্গ্যানস, অর্গ্যানস ভীষণ ইম্পর্টেন্ট। আমরা আমাদের সোমা বা বডিতে বোনস দেখতে পাচ্ছি, সেই বোনস তোমাকে অ্যানাটমিক্যাল পজিশনে ধরতে হবে। আমরা তোমাকে বলি হোল্ড দ্য বোন ইন অ্যানাটমিক্যাল পজিশন এন্ড টেল মি ইটস সাইজ। ফিমারটা ধরিয়ে দিলাম, অথবা টিবিয়াটাকে ধরিয়ে দেবো, ফিবুলা ধরিয়ে দেবো, টেরাস ধরিয়ে দেবো, ক্যারকিনিয়াম ধরিয়ে দেবো, বলবো হোল্ড দ্য বোন ইন অ্যানাটমিক্যাল পজিশন এন্ড টেল মি ইটস সাইজ। তাহলে ঠিক ঠিক বোনটা তোমার শরীরে আছে এইরকম ভেবে ডান দিক বা বাঁ দিক কোন দিকে আছে সেটা ফিচার থেকে ডিটারমিন করতে হবে, করে তোমাকে সাইজ বলতে হবে। পুরো তার আর কিছু নেই, টেক ইওর টাইম, টাইম নিয়ে দেখে বলবে। এবার তুমি যখন পরীক্ষা দেবে 2 মাস টাইম তো আমি তোমাকে অ্যালাউ করবো না, বুঝেছো? অল্প সময়ের ভেতরে তোমাকে দেখে তাড়াতাড়ি করে দিতে হবে। এবার অ্যালটেড টাইম থাকে পরীক্ষায়, ভাইবা পরীক্ষা খুব তাড়াতাড়ি দিতে হয়, তাই বেশি সময় পাবে না, অল্প সময়ের মধ্যেই, সেই জন্য ভালো করে পড়তে হবে, আর তাহলেই বলা যাবে। এবার হচ্ছে অর্গানস। এবার অর্গানস দেখো, আমাদের হিউম্যান বডি ইজ বাইল্যাটারালি সিমেট্রিক্যাল আউটওয়ার্ড। আমরা যখন বাইরে থেকে দেখছি তখন কিন্তু বাইল্যাটারাল সিমেট্রি মেইনটেইন করে। তাহলে অন আইদার সাইড অফ দ্য মিড লাইন, হ্যাঁ, একই রকম, একটা রাইট হাফ আছে, একটা লেফট হাফ আছে। কিন্তু ওয়ান্স উই আর ইন দ্য ইন্টেরিয়র, আমরা যে মুহূর্তে ইন্টেরিয়রে ঢুকবো, ইন দ্য ট্রাঙ্ক স্পেশালি, ট্রাঙ্কে যখন আমরা ইন্টেরিয়রে যাচ্ছি, হেড থেকে নয়, কিন্তু ট্রাঙ্কে যখন আমরা ভিতরে ঢুকবো, আমরা দেখবো দেয়ার আর সাম আনপেয়ার্ড অর্গানস। ঠিক আছে, খুব ইম্পর্টেন্ট আনপেয়ার্ড অর্গান কি লিভার, খুব ইম্পর্টেন্ট আনপেয়ার্ড অর্গান কি প্যানক্রিয়াস, খুব ইম্পর্টেন্ট আনপেয়ার্ড অর্গান কি স্টমাক, খুব ইম্পর্টেন্ট আনপেয়ার্ড অর্গান কি স্প্লিন, খুব ইম্পর্টেন্ট আনপেয়ার্ড অর্গান কি ইউরিনারি ব্লাডার। ঠিক আছে, মূত্রথলি, ইউরিনারি ব্লাডার, রিজার্ভার অফ ইউরিন। ঠিক আছে, আনপেয়ার্ড অর্গান ইউট্রাস। ঠিক আছে, তাহলে so many unpaired organs are there along with paired organs. তাহলে paired organs এর ক্ষেত্রে তোমাকে anatomical position, site determination first duty will be anatomical position, holding the organ in anatomical position, telling its site and then the features. তুমি কিন্তু ফিচার্স আগেই বুঝে নিয়েছো, অ্যান্টিরিয়র, পোস্টেরিয়র ইত্যাদি, তারপরে ফিচার্স ডিটেইল বলতে হবে, জিজ্ঞেস করবো আমরা পরীক্ষায়। আনপেয়ার্ড অর্গানের ক্ষেত্রে তো সাইট ডিটারমিনেশনের ব্যাপার নেই, তাহলে আনপেয়ার্ড অর্গানটাকে ঠিক ঠিক ধরতে হবে। তাহলে ওয়ান্স ইউ আর আস্কড টু হোল্ড দ্য লিভার ইন অ্যানাটমিক্যাল পজিশন, লিভার তো একটাই আছে, ইউ হ্যাভ টু কিপ ইন মাইন্ড মোস্ট অফ দ্য লিভার লাইজ অন হুইচ সাইড, মোস্ট অফ দ্য লিভার লাইজ অন দ্য রাইট সাইড অফ দ্য অ্যাবডোমিনাল ক্যাভিটি। ঠিক আছে, therefore since most of the liver is on the right side of the abdominal cavity, it will have a conspicuous right surface. একটা রাইট সারফেস সুন্দর আমরা দেখতে পাবো, ওরকম কিন্তু ডেড সারফেস বলে কিছু নেই, বুঝেছো? তাহলে একটা রাইট সারফেস দেখতে পাবো। তারপরে রাইট সারফেসটা দেখার পর আমার কি দেখতে হবে, সামনে পেছনে করতে হবে। যদি আমি পেছন দিক থেকে সামনে দিয়ে দিতে পারি, সামনে দিক থেকে পেছন করে দিতে পারি, না হবে না, বুঝেছো? উল্টে দিতে পারি, নিচেরটাকে উপরে, উপরটাকে নিচে করতে পারি। তাহলে এভরি সারফেস will have its features, will have features of its own. Every surface, superior surface or upper surface, নির্দিষ্ট ফিচার থাকে। Inferior surface, definite features will be there to recognize that surface as inferior. Similarly, posterior surface will have definite features which will help you to recognize that surface as posterior. Anterior surface will have definite features which will help you to recognize that surface as anterior. তাহলে এইভাবে বুঝে লিভারটাকে ধরতে হবে প্রপারলি, as if the liver is yours. বুঝেছো? তাই প্রত্যেকটা অর্গান, প্রত্যেকটা অঙ্গকে নিজের বলে ভাবতে শিখতে হবে। প্রথমে অ্যানাটমি শুনে নিজের বলে না ভাবতে শিখলে তুমি অন্যের লিভার নাড়াচাড়া করতে পারবে না, বুঝেছো? তোমার শরীরের মধ্যে যা আছে সেটা তুমি বার করে তোমাকে হাতে হাতে ধরতে পারো, বুঝেছো? কাজে মরার থেকে তুমি যা পাচ্ছ সেটাকেই নিজের করে গ্রহণ করো, বুঝেছো? অ্যাকসেপ্ট ইট অ্যাজ ইউজ। কাজে I will urge you to buy the books as soon as possible. কোথা থেকে তুমি জোগাড় করবে, that is not my duty. তুমি ask your seniors, they will tell you from where you can buy the bones. Bones কিন্তু কিনতে হবে, ঠিক আছে? bones, এই লং bones গুলো অবশ্যই কিনতে হবে, vertebral column কিনতে হবে, ribs কিনতে হবে, ঠিক আছে? সবই কিনতে হবে। সব কিনতে পারলে ভালো, ভাগ করে কিনতে পারবে। ধরো হোস্টেলে যারা রুম শেয়ার করছো, যদি মনে করো না আমরা পার্মানেন্ট বন্ডিং আছে, উই উইল নট সেপারেট, ডিভোর্স করবে না, হ্যাঁ, মিডওয়েতে ডিভোর্স হবে না। যদি এরকম হয় তাহলে পার্টনারশিপে কিনতে পারো, কারণ মঞ্চের অনেক দাম, ঠিক আছে? তোমাকে তো এই সমস্ত পড়াচ্ছে, ফাউন্ডেশন কোর্সে বলেছে বন্ডিং ইজ ভেরি ইম্পর্টেন্ট। তাহলে ডক্টর পেশেন্ট বন্ডিং এর অনেক আগে হচ্ছে। তোমাকে তোমার যারা ক্লাসমেটস আছে তাদের সঙ্গে ফ্রেন্ডশিপ ইউ হ্যাভ টু এস্টাবলিশ। আচ্ছা, ফ্রেন্ডশিপ 150 জনের সঙ্গে ফ্রেন্ডশিপ হবে না। Some of the guys will be your friends. Don't make others enemies. ঠিক আছে? Some of the guys will be your friends. Some will be very close, dear to your heart. Others will not. Don't make anybody enemy at all. কাউকে কিন্তু enemy বানাবে, ঠিক আছে? Make alliance with all. ঠিক আছে? Make alliance with all. আর রুম যদি শেয়ার করো, যার সঙ্গে রুম শেয়ার করবে তার সঙ্গে বন্ডিংটা তৈরি, যাতে করে মাঝপথে গিয়ে সেপারেশন না হয়। তাহলে বুকস কিনে পড়তে পারো একসঙ্গে। এখানে তো সব রকম সিটার আছে, আমি জানি 2 সিটার আছে, 3 সিটার আছে, আছে না? নাকি নেই? বলো, আমি তো জানি না, আমি তো অবস্থা থাকিনি। আছে সব রকম। তাহলে যেখানে যেরকম আছে, যদি 3 সিটার থাকে, যদি তোমরা মনে করো যে না আমরা একসঙ্গে থাকতে পারবো, 3 জন মিলে কিনতে পারো, ঘুরিয়ে ফিরিয়ে পড়বে, কোনো অসুবিধা নেই। হ্যাঁ, তাহলে আজকের মতো এখানেই থাক, 4:05 বাজে।